initiative funds, which is another complicated phrase for open science grants. So every year we run these sort of competition where researchers can get money for a new initiative on open science. Right, so now I've talked about what we did at the Neuro, but the second mission is throughout Canada, so right, the snowball effect for open science, and that goes through the support and partnership framework of TOSI, and this has about 3 different kind of levels, we could say. First is the seed projects, where we collaboratively build open science capacity in collaboration with other institutes. So that could be either exporting an open science platform or solution that we built here to other institutions, or to co-develop a new platform because there's a need that's not fulfilled. Second one is buy-in project. That's a bit what I explained that the Neuro went through at the very start. So through these, we provide funding to enable another institute to hire people dedicated to designing the open science approach and principles of an institution. And we also provide support, sort of knowledge from the Open Science Alliance Officer, which has experience with all of these different institutions already. And then once an institute has designed their open science principles, usually through a buying project, and have them approved by TOSI, we provide yearly funding and ongoing support through these institutions that commit to open science. So this is what the alliance looks like at the moment. It's not as extensive as what you showed a bit earlier, Claire. It's kind of jealous, but we're getting there. So we have 4 approved institutes in addition to the Neuro that's on the right there. You can see them listed. And there are 4 more that are ongoing buying projects, and so we should fingers crossed, see them approved in the next year or so. And what's great with this is that, well, yes, it means more institutes are committed to open science, but better than that is that we now have a network of institutes that face similar challenges in open neuroscience, and that we sort of paved the way for each other essentially by creating new platforms that others can reuse or just exchanging knowledge. Now, the biggest sticking point that the Neuro and the other institutes usually face in approaching open science is intellectual property, and mainly patents. So basically, that's a legal right to prevent others from accessing, sharing, or using information without permission. That obviously conflicts with the open science principles, the open barrier-free access to use and reuse of research outputs. And what we found for the most part at the Neuro is that the IP doesn't really matter that much or less than we would have thought, and that most researchers are not really interested because they're doing research so early on the discovery process that having IP at this early wouldn't really make sense, or it's even harmful for them to advance faster. IP can slow things significantly down. For instance, I'm sure some of you have dealt with their contract offices to have data and material transfer agreements drafted between lawyers, and it takes months. Another thing that came up in our own internal analysis is that most patents actually don't lead to a product, a service, or anything useful, but it costs tens or hundreds of thousands of dollars for basically nothing. And finally, IP is not even necessary for knowledge transfer because there's more and more solutions, alternative solutions that are more open. I'm at time. Okay, I'll go faster for the next few slides. So basically, this is just to show that the Neuro is kind of the more radical in its approach to IP. Basically, we say that people won't use IP or should not use any IP, but other institutions can have more or less liberal approaches to that with different, well, depending on their context and so on. Just like to spend a bit of time on that slide because that might be interesting for this conference mostly. So some lessons learned about our platforms here and what kind of worked in our journey. And I'll use the CBIG data repository as an example for that. So first, we found that seed funding is critical. First, to empower open science champions, right? You find someone that's really willing to do something cool for open science, throw a bunch of money at them, see where they go. It enables new collaboration that wouldn't have been possible otherwise. And it enables to test boundaries, push the limit of what can be done, spend time on something that you wouldn't have really spent time on otherwise, For instance, CBIG has spent a ton of time and effort to build their new sort of ethics and data sharing governance framework to enable to share super sensitive human data. But the thing is, seed funding runs out, so you have to find ways to make this sustainable, and I'm sure this will come up a lot during the conference, right? How to find money to make things sustainable. And something that we found this seems to work is through this institutional support to gather the community around a resource, get more people to use it, more researchers from the institution to use it, and get feedback, right? So CBIG started super small with a small cohort of patients with fairly restricted types of data that could be shared to that or material. Then through training the community to use it and to get more people aware of the platform, we got more and more data into it and more input from the community to use it. They asked us to grow it more, add this type of data as add this cohort of patients. And so now it grew sort of naturally with the actual needs of the community. It was big enough that they made a big ask to other funding agencies like Braid Canada, and got a big grant recently to expand it into Neuro VIP, which will then integrate more and more platforms from the Neuro, more data types, more patient engagement, which will grow it all again and again, and hopefully this sort of wheel will keep on turning. So that seems to have worked with us. Hopefully it can work with others. And that's a very last slide, and basically there's a lot of cool things that happen when you turn to open science. You get more funding from agencies and philanthropies. You get, to give your opinion on open science initiative across the world. You get to get new partnerships with industry and consortiums, and you get to attract young researchers that are interested in moving forward. That's it. Thank you.
I think maybe we have time for one question, if there is a pressing question. Otherwise, we'll move on, and maybe you have time to think, and you can ask Gabrielle during the panel discussion. Okay, so if we're good, Tom Durkin from the Early Drug Discovery Unit here at the Neuro will tell us a little bit more about his experience with open science in his platform. Thank you, Tom.
Cheers. All right. Good afternoon, everybody. Welcome to the Neuro. I know for some of you, you've come very far from like 2 floors down. Others, you've come from much further away, so it's good to see you all. Got to admit, like, it's going to be hard following Gabrielle. Like, I know he actually managed to give a talk with someone else's slides. I actually have to try surpass him, and these are my slides, so hopefully we'll even do a little bit as good a job as he did. So I'm actually here, my capacity as director of what we call the Neuro's Early Drug Discovery Unit, or EDDU for short. And I'm going to talk a bit about how we use human stem cells in both our discovery and translational work. And so Gabrielle's really talked about open science at the Neuro. I'm going to give you kind of an insight into how we work as a platform. So what we do is, everything we do is iPSCs. It's a second acronym. It's induced pluripotent stem cells from human patients, and we use them to make any type of brain cell. The patients that come through the Neuro, we're collecting their blood in an open and consensual manner. This then gets sent into CBIG, and from there we're able to then reprogram it into what we call stem cells, and from the stem cells they're then used to make neurons, glial cells, or other models that are more complex, even like a 3D brain, organoid, or neurosphere. So we have 3 main goals within our group at the EDDU. One is to make the iPSCs accessible to all, so companies, academics can access them, work with them, use them in their projects, to train users on how to actually work with these, and to actually work with both academics and industry to really unlock the potential of these iPSCs for discovery and translation with open science at its core. So we're kind of like an old, young unit, I guess, depending on how you cover age. Like, we started pre-COVID, we aged 50 years during COVID, and now we're 8 years of age technically. I actually started my postdoc with Ted Fon in 2007, so I'm coming up to 20 years at the Neuro in a few years, where I'll get hopefully a chair or a clock or whatever they decide to give you when you've been here a while. But when I started working with Ted, we were studying Parkinson's, and we were really working with HEC293 T cells, HeLa cells, mouse models, but we wanted to work with a human disease and really not actually have access to human tissue. So that's really where Ted kind of decided, let's try found a group how we can actually have human stem cells at its core, and Parkinson's was the first thing we did. We started in 2014 with seed funding from Brain Canada, and gradually over the last 8 years have grown it into a group which I now lead, where we have about 35 to 40 people, 25 full-time staff, and about 5-6 students with undergrad trainees that vary over each year. What it is it's kind of this circle here. So at the top of it, it's really CBIG. So CBIG is Clinical Biological Imaging Genetic Repository. We can operate really without access to the human patients. We're able to get the stem cells from them, and we're then able to reprogram them into these iPSCs. With the stem cells, we can actually use them and make them into cell types of interest to us, or we can actually use CRISPR genome editing technology to then knock out genes, to introduce mutations, to actually correct mutations as we would need. And then from there, we actually then use it for discovery projects to make cell types that people would want. Carl's going to talk about Icarus in a few minutes, and I'll get into that shortly. You can then use it just for understanding, why is a disease happening? Is there a specific phenotype that I can build a high content assay around it, and then ultimately then bring it in to a therapeutic, and then gradually test it in a preclinical model, and hopefully then the goal would then be to take it into a clinical trial here, and then hopefully then have success with the patients. So this would be kind of the older model, and we're trying to see, can we start to update it a bit with more complex models where you still go from 2D cultures, so more pure neurons, pure glial cells, you get a kind of phenotype, and then you bring it into a more complex human organoid model, and then gradually then you could take it into a preclinical rodent or an animal model as well. And this is kind of part of a project we're actually doing in partnership with Toronto, UBC, and Western called Trident. So we're divided into 4 main units. Everything we do is human iPSCs is done at the core. We don't work with animal models. We don't work with immortalized cells anymore. The main goals, what we try to do is we try to have 4 things we do. One is to kind of have a group dedicated to generating, phenotyping, editing cells, kind of a discovery and screening group, a 3D kind of tissue engineering group, and a training and outreach group. This is a big part of how we kind of bring the open mission within to our group. So we're centered around 3 main diseases. We started with Parkinson's, and we try to match everything with both 2D cell types and 3D kind of complex models. When it came to Parkinson's, dopaminergic neurons were the cell type lost, so it seemed logical to work with them. And then we gradually then got into making these complex midbrain organoid models where they would have the neurons and also then all the other subtypes to go with it. Gradually we got into ALS, where we were making motor neurons, astrocytes, microglia, and then you would start to make these kind of complex 3D motospheres or 3D kind of motor astrospheres. And then gradually now, more recently, into kind of neurodevelopmental, neurodivergent disorders like autism, intellectual disability, where we would then make cortical neurons, oligodendrocytes, to then actually have the 2D cells and then have kind of cerebral organoids, forebrain organoids, or even cortical spheres for then modeling it. One of the things that we try to do is, as Gabriel mentioned, is since we're open science, we actually try to make a lot of what we do open and accessible, and it does take a lot of work to do that. Over the years, actually, we've established a database of 20, and it's probably a bit more, over 20 open protocols. This means that we actually have written the protocol up in a standard format, and we've put it online in Zenodo, where it has a DOI and people can download it and access it whenever they want to. So far, it's about 12,000 views or downloads, and that was probably since I last looked at it in July, and it could have gone up more. But the other thing is, since COVID, is people would often get the protocol and then it would come train with our people within the lab. With COVID, we couldn't actually bring people into the lab for 2 years. Oops, sorry. Move that out of the way. I'm still making sure you're all awake. I know it's still Monday. I did get vaccinated on Friday twice, so I'm still recovering a bit. So the other thing is how to actually make this stuff accessible. And that actually led us to build a kind of video protocol development team within our group, where we actually have cameras and everything. We got for a few $100s, it goes into HUD, and then we can actually record all the various kind of protocols that we do. We've now put out about 35 plus videos. Anyone can download them, you can view them, and we've started to put them into different languages because first off, A lot of people look at them in English, but we've started to put them into French, German, Portuguese, and gradually we'd like to get it into other different language types. So we've only been going about 8 years. We started with 0 people. We're now at about 35 plus team members, goes up or down depending. We've trained over 300 users, 80 plus collaborations. It's about 140 plus IPSCs we've made. That's a little outdated. We just did the numbers last week. It's close to 160. And we've now put out close to 50 plus both video and written protocols. This has led to a lot of different 45 plus open access peer-reviewed publications, industry partnerships, and a lot of views. So in a way, by putting stuff out there, people then get to know you, and then they come to want to work with you, and then you get invited to be on a lot of different projects. And yeah, as I said, an in-house open science catalog of 160 IPSC, so that's out of date. To really kind of get buy-in from people is you have to make sure a lot of the stuff you do has kind of reproducibility, and you've got QC at the core of everything you do. When you work in a platform, people question you all the time. If an experiment works, it's because of them. When an experiment fails, it's because of you. We often find that's the way. I'm sure you've seen it yourself. So that's often why we always try to have good IPSC QC, where when we make a cell line, we try to have the karyotyping done. We used to do it at Sick Kids. We've actually now brought it in-house. We've been able to recently hire a pretty talented cytogeneticist. So she's been able to actually bring this in-house for us. Saves us a lot of money. But we do the SDR profiling at Sick Kids, genomic stability test we have to do, ICC, also have to do some of the other tests as well, like mycoplasma. That's done on a weekly basis. And this all goes into CBIG. So you'll actually, if you want to know more about it, we have a poster tomorrow. Nico and Taylor are going to be presenting it, so you can actually see it and actually talk and ask them questions about how do you want to access iPSCs, what's the process. It's actually pretty straightforward. All you need is an ethics agreement to work with stem cells, and then you need a project and a kind of an open science component at its core. So once you have the ability to make the cell types, then you can actually use them for different projects. So I'm going to talk a little bit about how we kind of apply this and how we work as a group. We work a lot with academics. It's kind of, we're in an academic institution, so we have to have an academic mission. And so I like to we've done a lot of collaborations, but this is one of the favorite projects I like to talk about. My favorite PI sitting right there, and Peter McPherson. And then Armin's actually one of my favorite grad students we've worked with over the last few years, because in a way, we had to provide some of the cell types plus some of the synuclein fibrils to actually gain insight into why and how is synuclein getting taken up by these neuronal cells. It led to a project and a publication in Cell Report, and now we've actually got the second publication coming out soon, or hopefully when we get past Reviewer 2 and Reviewer 3 and Reviewer 1 into whichever journal we're into next. But it kind of just shows that by working together with the academics and with us, we can kind of actually make some really cool science happen once everything is put there. The other thing we can also do is you can really start to actually apply this stuff into screens. So in a way, we were able to develop the kind of insight into the academic and kind of just discovery, but you can actually take some of this knowledge and actually build a high content screen. Andrea and Wolfgang are sitting at the back. Probably most of you know Wolfgang. I know he's been at many of these meetings over the years, but they're going to have a poster talking about our automation, but you can take the stuff with the cells and build a high content assay, screen for compounds, screen for therapeutics, biologics. And some of the examples that we've done is actually with synuclein uptake. You can label these aggregates with fluorescent tag. You can then quantify how rapidly it gets taken up in neurons and in other cell types. And then you can actually start to build a high content screen where you automate the seeding of the cells, you automate the pinning of the compounds, the treatment of the cells with the compounds, and even then once you fix them and image them, you have a whole automated analysis pipeline. Just to show you an example here with the Prestwick library, it's about 1,500 compounds, and then gradually you can find 2 or 3. These are tool compounds we found that let us then rapidly kind of show proof of concept that when you add them in, you can really then block synuclein uptake pretty well within it. And they're going to have a poster. You can certainly ask them more about that. The other thing you can do is you can also take it into other cell types. It's not just neurons. Everyone loves focusing on neurons for CNS disorders, but there are a lot of other cell types like microglia. This is actually a publication that was, it's actually the preprint here is actually just accepted in Brain recently. So while I run a platform, I also run a scientific group, so we still have to produce scientific knowledge to still be kept in a job. And so this is actually my student, Marie-France, was able to kind of take some of the knowledge about cells, microglia, and show that the receptor MERTK was actually kind of helping mediate this uptake of synuclein into the cells themselves. So the other thing we do is we try to see is how can we take stuff from a very simple kind of 2D model and build more complex 3D models. And this is more stuff that when you have a phenotype or you have a kind of an answer, then you can really start to kind of take it into a more complex model. And since this is more kind of related to brain organoids. And so why go from 2D to 3D? Well, for instance, when you're working with a flat culture, it doesn't really have the same interconnectedness that you would have when you'd be working with brain tissue. And that's kind of what you get with the brain organoid. It's quite scalable, so you can actually make hundreds of these, and you can actually grow them for a long period of time, and they're easy to maintain. Media change once a week, and it almost becomes like if you're working with animal models. You start to set them up and stagger the cultures in a way that you have hundreds growing at any one time, but you also get a diversity in cell types. You've got neurons, glial, oligodendrocytes, everything you would need, and they're customizable. You can make them based on how you would want, and the goal really is to see can you then take them and apply them to test therapeutics. So how do you make them? So when you have iPSCs, you actually culture them in flat culture dishes, and then you start to aggregate them, put them together, incubate with Matrigel, and then you kind of put them into these shaking, and then you start from this small embryoid body here and go to a 2 to 4 millimeter size organoid. This is what it looks like here after about a month or so. They're translucent, but if you add L-DOPA, they actually stimulates the dopaminergic neurons to then become active and go brown. So we were able to then make these and actually gradually show through a report by V. Mohammed and others that once you had a Parkinson's mutation, you had excess synuclein, you actually could recapitulate a number of features of Parkinson's with actually a 3D brain organoid model. You could show that with increased synuclein, you had more aggregates that would form, you would have a decrease in glial cells over about 150 to 200 days, and you would actually have a decrease in dopaminergic neurons. But it took a long time. It's not like something that takes weeks, this takes several months to get. So in a way, we were able to show a first model that we could do this, and then gradually now we're starting to see how we can extend this, expand it into other disease mutations, and then gradually testing of therapeutics over the next few years. And this is really where industry has come in. We actually just startups around, we don't just work with large pharma, we work with a lot of startups of technologies that then we need to see is can we combine this technology and see if it would work. And you'll see here, this is actually a bioreactor of organoids, 3 months old, just shaking around. This is actually used with company Inuvia, where we're able to kind of work with them to form these discs, where we could take iPSCs, seed the suspension, make hundreds of these EBs, put them in a bioreactor, and then we just would grow them. And then you could do multiple flasks at any one time. You could stagger them, you can take them out when you would need, and they can grow up to 18 months. And it really let us kind of scale a lot of this up. And once you have them, you can really now start to build a patient profile for iPSCs, where you go from the patient down to the iPSCs that you would need into more of a kind of a simple 2D cell type, neurons, glia, and then into a more complex organoid model. And from there, this is really what the imaging people come in. They help us image this stuff. What do the cells look like? How do they look like morphologically? And then how do you then reconstruct a 3D model? These are not simple things to do, and it's taken many years to build the workflows to do it, the imaging analysis pipelines to gradually now where we do it. You have all the data, and now you have to get going. And so I'm going to start wrapping up shortly. So one of the things to do, the last thing is then how does this look like as a kind of a single cell? That's another feature we're trying to do, is single cell phenotyping. So just to kind of come to a thing, this is where we're at now. We're trying to really, we've gone from the patient to the stem cells, to building assays, to building all the different cell types, both simple, complex. And even when we say 2D cells are simple, they're not that simple. There are actually a lot of difficulties that go into it. But gradually the goal is how do you combine it towards finding new therapeutics? And so hopefully we've kind of, in the time we've shown you is through our goals within our group, it's really make our iPSCs accessible to all. If any of you will like iPSCs, please talk to us. We're happy to kind of see and work out a way to do this, to train users. We've trained groups both in McGill, we've even trained people in Manitoba, we've trained people in Saskatchewan, Alberta, even in other places in Ontario too. It's not just restricted to the West. And then also then working with academics and industry to really unlock the potential of these cells. They really can allow a lot of discovery both in the CNS field, but even in the non-CNS field as well. So thank you for your time. That's our QR code if you want to learn more. 3D sphere, just to show you what they look like when you can image them. I always like that. And so I'll take any questions, comments, or I'll go to the next person. But If Peter has a question, I could take it, I guess. Yes.
So, how, in terms of open science, how does it work with industry when you're doing drug screening? I mean, do you just give them an advance look at the data, or is it all completely open and then just, you know, how?
Yeah, so it's kind of like, how does it work with industry? So we kind of work on an 80-20 model. We're always trying to find, like, what's the open component? Like, so we're not like, you invest money, we do the project for you, we give you the results, nothing is made open. So we've worked with the INP office, OSR, so that any of the contracts we do has specific open science language that money invested has to lead to something tangible. Publications, cell lines, and assay. So often when they're investing in an assay, we get the assay and we can apply it across all our other projects then. The only thing that they really get in return is sometimes they're just trying to see is there kind of assay or is there a phenotype there? And often it's more, can we reproduce what's already in the field? Sometimes we find the publications that have already come out doesn't work out, and then the project won't move forward. But if it does, then it's a case of how do we automate this assay, and then how do we test specific compounds. Often we'll add tool compounds that we can then add into a publication, and their compounds will be on the side. And that's really what they're looking to get is, if they have a compound, will it work in a human model, and then maybe potentially then take it then onto something more advanced.
I guess their real IP comes way down the line anyways, right?
Yep.
What the way they do the transfers.
Yep. Maria?
In terms of your patient consent, at the very beginning, you were showing that you take blood and then you sort of said you can do many, many things with that. So how does your patient consent form look like so that you can actually do so many things with your patient samples?
So Nico's there in the back. So this might be, so Nico, how does the patient consent form work and why can we do so much with it?
So you pretty much try to think of every possible thing that you could do with that, and you write it down. Okay, thank you.
There is a microphone in the
Just
two
at the
aisles
back.
too. Put on mine. I'll just scream a bit. Since it's a platform, there is sort of usually a bit of a business aspect. I was wondering, how does it work? So somebody comes and says, I'm interested in this. Can you get me a specific patient? You pay for the whole project, or you just essentially sell these organoids? Or just
Yeah, everything we do is customizable. It's like a
Everything.
plug and play. So often some groups will say, we'd like to make a CRISPR knockout. So we'll then figure out what the price is and give it that. Often other times it's more, I have an idea for a project, but it's going to take X amount of years to do. And we say, okay, well, let's look at how much personnel time is going to be needed, resources. We'll come up with a budget, we'll present it to them and then go, if you can't pay for it now, then we start figuring out ways we could find seed money, grant money. So a lot of my job is raise money. Like I'm almost gone from being a scientist to almost more, how do you hustle foundations and other people into giving you money to then leverage it into the next amount of money and the next. So it's always, every time when we get money, we try to seed it into more money and then that snowballs down. It's getting harder with inflation and high interest rates.
Yeah.
The money is becoming less, but we do have certain, like IPSCs themselves, if you want to buy a line, they have a very specific price point, but often.
So you do have certain line items, so to say, that have a given price. You order it, catalog, you get it.
Yeah, but
But-
that's done through CBIG. So CBIG sells, like if you want to get a vial of blood, a vial of an iPSC, we have a reduced academic price for the Neuro. There's a price for academics outside of the McGill network and other affiliated networks, and then an industry price. So we do price that, but for our projects, often we have to develop a project plan, then we cost it out, and then if it's with industry, we take it to INP OSR, we add the 40% that McGill wants, and then give it back, and if they sign off on it, then we can go ahead with the project. And there's often, it comes with milestones, so it's all contingent on do you hit your different stages to then take it to the next level.
Thanks. Kind of a follow-up question on what Maria asked earlier about the patient consent form. So you mentioned that you also have to catalog IPSCs. So is the consent form also kind of covers that part? Like once the IPSCs are generated, then it could be commercialized?
There is a commercialization element to it. We are in discussions with a number of companies about how to really license lines out of it. It's more complex than it. Well, it's like anything with commercialization. It's complex. We deal with CRISPR or iPSC or programming technologies. They're both complex things, but we are in one company in particular. We've kind of completed some of the hurdles, but I think when it comes to giving them to academics, not a problem. When it comes to other things, when it comes to commercialization, then that becomes a part of, well, what's our project? And then it gets analyzed by the tissue data committee, and then they'll explore it. Most of the projects that have gone forward so far, it's all gone and been approved. So we haven't hit kind of a snag where something's not been possible, but a lot of it just is we have to sit down and kind of look at all the complexities that go with the project itself. So it's a bit of a wishy-washy answer, I know, but sometimes these things are a little tough.
I think we'll have to continue at the panel because, yeah, it was a great talk, but we'll keep the discussion going a little bit later. Thank you, Tom. So Nadia is going to tell us about or share a case study on utilizing the OSF for reproducibility. Thank you for being here.
Thanks. You'll have to give me one moment to just set up because I was also late in submitting my paper. Thank you. Yeah, I'm just trying to connect to the internet. I'm just trying to connect to the internet, but there is no other way for me to. Oh, here it is. Sorry about that, everybody. Okay, sorry about that. brief moment of pause. So I just want to thank Vidhu Sharma for bringing me here. I'm from the Center for Open Science. CNSP meeting is new for me, but so is the Center for Open Science. My position at the Center for Open Science is a strategic partnerships manager, so I'm often interfacing with consortia, institutions, research organizations, specifically librarians, research coordinators, data managers. So I interface with a lot of different types of folks, primarily at institutions, but also engage a lot with researchers and try to understand how researchers are utilizing the platforms that we build and the services that we provide. A little bit of context for those of you who don't know about the Center for Open Science. We're a 10-year-old or nonprofit organization. We're based in Charlottesville, Virginia, in the U.S. We emerged out of a partnership with the University of Virginia, UVA, and the lab of our now executive director, Brian Nosek, who's a social psychologist. You saw an image that he provided by one of our speakers, and I'll be returning to that image because it's a big part of what we do at the organization. So at the Center for Open Science, we work with researchers, librarians, as I said, journals, funders, government agencies, policymakers, journalists, research administrators. For the past 10 years, our mission has been to support open scholarship across the research life cycle. So from the very early stages of planning or coordinating or collaborating to publishing of outcomes. Our sense is that to scale the adoption of open practices by researchers, we have to provide infrastructure that makes it possible to engage in open science. And so I think it was Gabrielle who was talking about the infrastructure that has been built at the Neuro. We also build a much more generalist kind of software infrastructure for researchers. We also offer solutions to journals and funders and institutions to try to do things like nudging incentives to make it more desirable to practice open science. And we also promote a policy framework for stakeholders to require open practices. Ultimately, kind of our end game and our mission is that we, want a future where scholarly communities are engaging in all of the different aspects of open scholarship, but we recognize that that's difficult, and every community and every institution is going to need to approach open scholarship in their own way. So some of the products and services and research and policy that we do, you can see here. We have a lot more information available on cos.io, which is the Center for Open Science's website. If you're interested in diving in any deeper, we offer products like OSF Institutions, which is for research administrators and libraries to understand how their research community is using our software infrastructure platform. We create badging frameworks, and we have something called Top Factor for journals to incentivize open research practices. We create and have and support white-labeled preprint services. We support registered report workflows, and we also provide openness and reproducibility trainings. One thing, I don't know how many of you are familiar with the OSF or have an account, but highly recommend you just make one today. It takes 2 seconds. It's free for all researchers, and it gives you a kind of ability to play around with some of the workflows that I'll be talking about today. So the Center for Open Science, in addition to providing all these different types of products and services, is also engaged in dozens of multi-year research collaborations to try to underscore and test our theory of change. So if we make open science practices required, if we make them rewarding or normative or easy or just possible through our software infrastructure or through any kind of infrastructure, what impact will those practices have on the reproducibility or rigor of research? And so a lot of the research that COS has conducted over the past decade on reproducibility and rigor has utilized the OSF platform that we built at the organization and that we support and continue to build. And we've done that to try to test the effectiveness both of this platform that we've built, but also of the open practices that the platform supports. So things like data sharing, collaboration, registration, pre-registration, and registered reports. So what is the OSF? You might be extremely familiar with this already, excuse me if you are, but I'll just repeat it because it's often a conversation that I have every day with lots of folks who are very familiar with open scholarship, but not necessarily with the platform. The OSF is the kind of powerhouse that supports our research, but it also supports over 500,000 researchers who utilize the platform. It's free, it's open source. It functions as both a generalist repository and a publishing platform, in addition to many other things. We've got to let them be. And the OSF also provides really powerful project management workflows for researchers and research communities, as well as collaborative or collaboration workflows. So as a user on the OSF, depending if you're on the product team like I am, we call them users, but they're also researchers. They are also many other types of people with different types of roles, but as an individual on the platform, you can do primarily three things. You can create projects in a project workflow, you can create registrations in a registration workflow, and you can create preprints in a preprint workflow. And these workflows are integrated into a lot of the life cycle research projects that the Center for Open Science has embarked on in collaboration with other research groups over the past decade. And just something to flag that I'm not going to really dive too much into, but is a really big part of what I do at the organization and is also increasingly needed to meet policy mandates and recommendations in the US, North America, Europe, and elsewhere around data sharing. Is that we have workflows for organizations or administrators. So the ability to moderate and curate research and workflows within a community to support the visibility of research or workflows in a community or at an institution, and also to engage with impact metrics around who in your community is doing what on the platform. So I've talked a bit about kind of our theory of change and meeting researchers where they are. The OSF workflows, that some of which I've just described here, allow for meeting researchers or research communities wherever they are in the open scholarship landscape. So it allows for discovery and sharing of scholarship at all different stages of the life cycle. So project workspaces and allow for kind of planning and collaboration and browsing and discovery at very, very early stages. Whereas registration workflows and registries, OSF registries, allow for research to be conducted openly and explicitly with materials openly available and archived. Preprint workflows come a little bit later in the life cycle where outcome reporting can be linked to both registrations and projects. So just a kind of an example of how this actually looks and what it is on the OSF. So this is an example of a project on the OSF. They're a really excellent place for teams to show their work, refine ideas. They're centralized workspaces where data, materials, information about a project can all be organized and connected. Files can be uploaded and versioned for tracking purposes. You have activity logs, different types of components with different privacy levels and settings, as well as different licensing. Collaborators can be added and removed at any time, and all public projects receive a DOI, or we have a DOI minted for those. A registration workflow, which might be more or less familiar to you depending on what domain you work in. The registration workflow is a very common one within, or increasingly common in social sciences. This is a time-stamped archived object on the OSF. So a project on the OSF is more like a flexible wiki space for sharing and collaborating. A registration is archived. It can never be deleted. It outlines the research and analysis plan prior to starting a study. A type of registration workflow called a pre-registration workflow also allows researchers to specify research plans in advance of conducting the study, and these cannot be deleted once they're published. You can update them. You can see the little update tab there, but these will always remain on the OSF, unlike projects, which can be removed or added at any time. Registered reports, which is a publishing format. Just a quick show of hands, because I'm not sure. Is anybody familiar with registered reports? Oh, awesome, great, just a few people. Okay, so registered reports is a publishing format that's increasingly accepted by hundreds of journals that allows for peer review prior to data collection. And so utilizing the sort of registered report framework is possible on the OSF and using the registration workflow. In the publishing and research format and workflow that I've outlined here about registration, pre-registration, registered reports, it can help to eliminate a variety of questionable research practices, including low statistical power, selective reporting of results, publication bias, and it allows flexibility to report serendipitous findings even though you're discussing your study before it is conducted. A couple other features of the OSF workflows. So again, these are registrations, projects, preprints. You can collaborate with anyone in the world. You can fork projects, create project templates to be utilized in a specific community or for a specific project. You can share your data. You can set fine-grained permissions on all different parts of a project and license your projects, as I said, in a flexible fashion. We use fair metadata, and we have a really robust OSF metadata schema, and we also work with different communities to try to integrate their metadata needs into the platform. You can also, as I said, mint DOIs for public projects, but we also mint DOIs for registrations as well. And you can sync your OSF content with ORCID records. Because we're open source, if you have the capacity to, you can utilize our API to propagate data and metadata downstream in all different types of ways. I'm going very quickly through a lot of these kind of features, but I just want to flag that if you do have any questions about how this has been utilized by different communities or researchers, please come find me and I'm happy to talk about it. So who uses the OSF? We built this platform for researchers, but a lot of different types of people utilize it. As with any software infrastructure, you have an intention and then you set it out into the world, into the wild, and people are using it in lots of different types of ways. This was just a qualitative survey we did of around 5,000 of our users. We have around 500,000 total users. A lot of users come from the social sciences, but we're seeing year-over-year expansion of disciplines as well as roles. So a lot of our users are researchers, early career researchers, graduate students, undergraduate students, but we have a significant number of research administrators who work, as I said before, in research departments, but also in libraries at different institutions. One thing that is kind of interesting that I just want to flag and kind of relates to our theory of change again and kind of what we're trying to do and what we haven't yet done, not just as an organization, but as part of an ecosystem of open scholarship, is it's interesting to note that a lot of people are utilizing our platform in lots of different ways across the research lifecycle. So they're using projects, they're using registrations, they're using preprints. But the way in which most people are engaging with all of this open data and open material is that they're just really looking at outcomes. Not entirely, but a lot of what people want to see and what they're accessing and downloading are preprints. And so this isn't sort of special to the OSF. We see this across lots of different types of platforms and repositories, but it's just something to keep in the back of your head as you think about kind of how to create incentives and norms at your organization. That this is a really big piece of it that we still have to hack away at. What is the data actually being used for? How is it being used? And who is accessing it? So how does the OSF, this platform I'm talking about here, this kind of generalist repository, project management workspace, tie back into the research that we do at the Center for Open Science? So I want to hone in on 2 studies, the one that was conducted over the course of 8 years with findings published in 2021, and one that was conducted over 6 years with findings published just last week. So these are the Cancer Reproducibility Project, which launched in 2013. And this was a collaboration between the Center for Open Science and Science Exchange, which is was the world's first online R&D marketplace that wanted to advance scientific discovery by matching projects with expertise. This project was supported by Arnold Ventures. And more recently, the Reproducibility of Social Behavioral Findings study, which was results of which were published a couple of weeks ago. And this was led by PIs from 4 labs at UCSB, Stanford, Berkeley, and University of Virginia and the Center for Open Science. So the reproducibility crisis is not a new problem, but it's helpful to note that when COS launched the Cancer Reproducibility Study in 2013, this question was very publicly, hotly debated. This was from a Nature survey of about 1,500 researchers from 2016 who were pretty adamant about the fact that there is a crisis and that they have encountered the crisis themselves with their own research and also with others' research. So the approach of the Cancer Reproducibility Project was that there would be a systematic sampling of 53 high-impact preclinical cancer biology papers from 2010 to 2012. These excluded genomics, proteomics, high-throughput assays, and the primary outcomes of the project were summarizing the process challenges for just testing replicability and also a meta-analytic summary of statistical outcomes. Most of the papers based on those selection criteria were from high-profile journals like Nature, Science, and Cell. And from 53 papers, a total of 193 experiments were selected for replication. The team designed replication experiments of key findings from each paper. They reviewed the methodology, they requested information about protocols from the researchers, and inquired about the availability of reagents, and then appropriate expertise for actually conducting the experiments was sourced through this Science Exchange marketplace. You might be familiar with this study since this was published a few years ago, but I think it's relevant to this new study that's just come out. A few key takeaways from this experiment: only 2% of these experiments had open data, and after repeated requests, 16% shared raw data. Summary data were shared for 14% of the experiments. Nothing was shared for 68%. And statistical analyses were reported for around 40% of the experiments, but when the outcome of the analyses was reported, it wasn't clear at all what statistical test was used in about 20% of the experiments. Code wasn't shared for the majority of experiments, and the majority of experiments also needed some to a lot of clarifications. And so out of these design studies, only 87 were conducted and only 50 were completed. So all in all, what were the barriers to replicability here? There was a lack of transparency, a lack of accessibility of methodology, a lack of sharing original data and reagents, lack of communication for obtaining needed information. There were a lot of unexpected challenges with protocols during experimentation, and a lot of resource challenges around cost, time, and uncertainty. So the takeaways from the meta-analysis here: replication effects were much weaker than the originals. There's a lot of data here, multiple papers. You can find all of them here, so I'm not going to go into them too much, but I would say it's a simplification to say there was room for improvement. But the positive results in general were half as likely to replicate as null results. So given these possible failures, there were some pretty clear, oops. I've lost my little. Sorry. So given these possible failures, there were some really clear takeaways. And one of the big ones is improving transparency and accessibility of methodologies, the sharing of original data and reagents, and just increased communication between researchers. These are all clear needs in the process of reproducing research. And so these, the OSF was a sort of key part of documentation of the Cancer Reproducibility Project, and utilizing all the open scholarship tools that I mentioned at the start of any research project could also improve reproducibility and rigor of other projects as well. And so that was the reason why some of the future projects that we engaged in terms of understanding, you know, are these practices impactful for reproducibility, why we engaged in them. And most recently, this paper, which you can also find here through this QR code, which was published a few weeks ago, we had 4 research groups that devised and planned totally new separate projects. So the idea here is, let's try to figure out, can we utilize all of the kitchen sink of best practices of open scholarship and see if we can get some good results around reproducibility and make a kind of benchmark for best practices. Each lab carried out these projects. They used all the practices that are known to increase experimental rigor, which I've reviewed, so pre-registration, increasing sample sizes, and specifically open sharing of research materials. And when the research suggested an interesting effect, the original lab ran a confirmatory study. And then 4 candidates, new discoveries were selected by labs for this self-confirmatory testing phase. And then the other labs ran a repeat of these 4 chosen studies. So again, very quick summary of the effect here is that the results show that original findings could be replicated 86% of the time, which is a significant and significantly better than the 50% success rate that was reported by a lot of other systematic replication efforts. So I think this study shows that research in the field can be top quality if all of the right steps are taken. And again, this is, you know, might be domain specific, but I think is a helpful first step in benchmarking. The overall framework of the study, again, as I said, was to try to use best practices in open scholarship workflows and tools to create this kind of benchmark. Many of the best practices I've talked about today and in the research that we've conducted at the Center for Open Science, they're supported on the OSF. And we built the platform for that purpose, to try to continue to develop these types of features and workflows and make open scholarship practices easy, accessible, to researchers at all stages of the research life cycle. I think this recent study and many of the other research that we've done at the organization serves as a kind of proof of concept enabled by the OSF, and we hope to continue more into the future. And thank you all for having me.
Thank you. Since we're running a little bit late, I'm going to ask Tom and Gabrielle and Nadia to go to the front, and we'll start the panel discussion on the topic of open science and open science institutions. You guys are there? Okay. So maybe I have a question to start, maybe to Gabrielle. I don't know, since the Neuro, I guess, started, you said, in 2016 or so with the open science mission or certification, whatever you call that. Can you measure at this stage the benefits of that, or how would you measure the benefits?
Yeah, good question. That's something with open science metric is something we've been working on more intensively recently. So we already measured qualitatively somewhat, and that's kind of some things are difficult to do. So for instance, you know, the number of new recruits that came into the Neuro, there was quite a great wave of new high-profile new recruits that came in since then. But then, you know, exactly.
Can you really tell if they came because of the open science?
Exactly,
mission,
it probably played some role, but you know, so that's that. And And then there's more clear things that we can measure, numbers of data sets that get shared, number of papers that are published open access, number of papers that contain open code, open data, open name it. And so this is kind of in the works. We've already got the simplest measures, like the open access papers is the easiest thing to measure, and that is going up from year to year. But again, the whole world is moving towards open access, and so now we're working on benchmarks to try to compare the Neuro with another comparable group, which is also kind of tricky. But so short answer is, we do have qualitative indicators that it improves, funding, recruits, and open access metrics. Not quite.
Do you feel like the fact that other institutions are joining is also an indication that there are benefits there, or?
Well, that's clear success, right? Like at the start, I don't think it was clear or to predict how many institutes would be interested in joining, and so far we've had quite a lot of interest, and there's even more institutes that are outside of TOSI's framework, meaning either they're international, so not Canadian, or that are not in neuroscience focused, that approach us looking for funding or collaboration and funding for seed projects or buy-in projects, and sort of have to say no so far. So there is clear interest in that.
Cool. Does anybody else have? Yeah, go ahead. Oh, sorry.
You were there first. You should go.
Okay, thanks. Yeah, hi, I'm Heidi Ibrahim from ALS. It's a laser infrastructure here at INRS, close to Montreal. In our case, we try to push towards open access or open science in general, but for example, the data is less meaningful if you don't know the whole experimental setup and everything, so it's really hard for others to directly use it. In our case, it would be, for example, very nice if all the research could be published in open access journals, but then there's always this competition that you need to publish in high impact, not high qualityness, but high impact journals. And then these are very often not open access, for example, or if some of them are, you need to provide an incredible amount of fees to get it. I mean, I've heard recently up to $40,000 for a review of modern physics, which is completely crazy. So there is this kind of conflict which we have, because we want to make our data accessible, and at the same time we don't want to lose to get nice funding later on, because we want to publish in these high impact journals as well. So what would be like a vision of how to handle such a situation? Of course, if everybody at the same time would say, okay, we don't support these journals anymore, then this is perfectly fine, but I'm afraid this won't happen. Thanks.
That's for me, yeah. I can take it. Yeah, that's a tough one, and well, the answer, as you said, will come from multiple levels. So researchers can nowadays choose to, or have more options to go in different outlets that are open access and with high impact, more and more that are not as costly. Then funding agencies and universities, higher ups, as it were, are kind of slowly changing the incentives to get away from impact factor and more towards more useful measures for science. So that will go away eventually. Also, what my suggestion for McGill researchers is to use the eScholarship repository, which is sort of a parallel way to get open access without paying for the open access fees at the big journals that charge a lot, and most institutions have that nowadays. So it's an institutional repository. It's got the green open access path, so you can sort of publish your closed version in whatever journal, like Lancet Neurology, for instance, not paid for the open access fees, if they even have the option. But in parallel, publish a version in the McGill's institutional repository of that paper. And the peer-reviewed paper, not a preprint, because that's kind of important for funding agencies. It has to be a post-review and not a preprint that gets open access to be compliant. But so there are multiple approaches that seems to go in the right direction. and eventually we'll get there. Too slow, but that's my answer.
Do you have anything to add, Nadia, maybe to that, or Tom? No? Okay. Next.
Hi, Pierre-Jean Larco. Hi, Marie-Ève. And I kind of want to add to the previous question. The issue with open access is that indeed the tri-agencies are increasingly demanding that researchers put their stuff on open access. And it doesn't matter how little it costs. And that means that although you have great ambitions and you're putting forward systems to make things open access and open source, one of the ambition is that so it increases access, but ironically, because that open access costs a lot, and unless your university has a lot of muscles, whether philanthropic or institutional, there are researchers that cannot be on board. And some, especially early career researchers, will want to publish in high impact papers, even though this is no longer or getting not to be the norm. You still need to publish, not to perish. And so these are hard choices for researchers that I haven't met a faculty that doesn't want to publish open access, but I've met many that cannot afford it. How do you address this paradox between your ambitions and the reality, if I should say so?
Honestly, it's like the $1 million question if we only had $1 million. I think, I personally, I hate the publishing model as it is anyway, but it's true. You have to publish to keep your job. You have to publish to get promoted. I think what we've found in recent years is a lot of the foundations, like the Fox Foundation, TOSI, and many others, they've helped kind of bring in funding to actually help alleviate some of the costs. So I think in a way that's helped us we've been able to publish in Brain and eLife and many others where the costs have then come down a great deal. I'm a big advocate for the preprint, to be honest. I know it's not received as much attention, or people do it now. I always see it as kind of you place your mark on getting your work out there, and then this way it reaches a kind of wider audience while you're going through the 12 to 18 month review process almost, which seems to take for big papers. But I don't think there's like a silver bullet that's going to solve the problem just yet. It's always you come up against a funding barrier. Ideally, journals would say we'll bring the prices down, and then universities and other funding agencies will put more towards defraying the cost. I think maybe if CHR, NSERC and all those are able to put a little bit of extra where you could apply to do that, that would probably help as well. But that would be maybe ways to do it, but I wish I had the magic bullet to say. I know, what's it like in the States? It's even worse, the Wild West there.
Yeah, I mean, along the lines of funding, like what we're seeing too is in response to the NIH Office of Science and Technology Policy mandate, which is similar to the tri-agency mandate to share research or data. It's not quite a mandate yet in the U.S., depending on what agency you're getting your funding from, but there are a number of private funders who are now requiring all research to be made open, and so there's like an increasing sort of vice clamp on publishers, I think. And I just came from a big acquisitions library conference in Charleston in South Carolina, where the huge conversation was around when and how are publishers going to sort of release that grip, because it's putting libraries into a bad financial position in the US, who ultimately have to cover the costs of making their research communities research open, either in their own repositories or elsewhere. And so I think it's a multi-pronged kind of solution. But a big one in the US, because a lot of funding for research comes from private foundations, Simons Foundation, Sloan Foundation. we're seeing that they're requiring a lot of open access, and that is pushing things along. That's incentivizing things. So,
are they putting money towards that though? If if they're requiring publishing in open access journals, they're putting money towards that, right?
yeah, and they have the money, which is a kind of a positive thing in terms of changing the norm. HHMI has required their researchers to openly publish their work for a long time. Simon's Foundation has done the same, and they fund a lot of research initiatives and projects, and they're kind of driving the field in that direction.
Interesting.
Oh.
Another comment,
Uh,
question?
Sure. First of all, I want to thank you for championing this fight for open science, because this culture change is very much needed. You guys are very courageous for leading that charge, so kudos. And then I have a more practical platform management question, I guess, for Tom. So in your presentation, you showed us how there's like a complex, I guess, administrative system you guys need to have in place for coming up with agreements with Office of Research, with startups or big pharma companies, and then dealing with the financial issues of balancing your budget and having fee structures in place. Do you have dedicated administrative staff that handles that? And if so, how do you pay for that? Is it institutionally supported, or do you have to seek soft funding to continue that going? Because I know a lot, in my communications with the CNSP, a lot of platforms face the problem of not having the ability to have administrative staff, and it's constantly turnover because students or postdocs are running their platforms. So do you have any advice on how to run the management side?
So we were fortunate many years ago. Before we started, we actually did manage a lab for a year or two. So in a way, we got to learn a lot of the nuances of ordering, who to talk to in all the different McGill departments. And then in a way, when we kind of went and took over the group and led it, we kind of knew all the various departments of who was the person to help us get the ball rolling, get the templates in place. And once we got that in place, a lot of it's been a lot more streamlined. So I am fortunate that I do have a bunch of senior people within our team. Like they are research associates, but they often do a lot more work almost at the management level, from invoicing to journaling to a lot of the financial side. But McGill and the Neuro, they do help a lot. Like we do have admin coordinators that help with a lot of the different kind of requests that come in. With the OSR office, they've been instrumental. So we have a process. It's not that difficult. A lot of it, once you template it, it's a case of we write the research plan together. Often sometimes it's my team with the other group. I help cost it out, and then it goes to the OSR. They look it over, and then they run their standard research agreement template. And then it gets signed, and then it goes over to the awards, and then from there, we deal with finance. And finance, we pretty much have like daily emails with them that they oversee a lot of the stuff for us. I think because we bring in a lot of business, then therefore they help us with a lot of stuff. Claire's been a big kind of pusher in a way at McGill of to get the different things in place. For invoicing, we've got a centralized system now. I know we just adopted it at the Neuro for the microscopy course, so that's simplified things. 10 years ago, everything was in like paper format. Now for like timesheets and everything, a lot of the stuff is on Workday. So I think gradually there's been more and more stuff that's come in to help ease the admin burden, but then it still goes up because the more projects you work on, the more you have to talk to people, and sometimes it becomes a challenge to like manage your day. But I think we've been fortunate, like we have a bunch of people in the back row that have really been kind of in a way, the unheralded heroes that have kind of, they keep the group around while I kind of go off and sit in meetings or write emails or hide at home, as I was once told recently. And they're like writing grants, but they're like, you need to come in more. So we try to be in a lot more, but Gabriel sees it as well with CBIG and other stuff. Like the busier you are, the more successful you are, the more resources you have. I wish there was a magic formula, but there's not. It's kind of trial and error, and sometimes people yell at you, and then you learn from your mistakes, and then make sure it doesn't happen again. I'll just say one thing. It's like, I never managed running a platform. Like, to be honest, it was my worst nightmare, but 8 years later, it's gone okay. And, if I get to 10 years, I survived the pandemic, a lab closure, and, all the other stuff that's gone with it, like, we'll consider that we've done okay.
Okay, great, thanks.
Hi, my name is Chloe, and I have a question, I think, for more all of you, because it's kind of really open discussion. So I never thought about science more than open since I am a child, but I think at the time where we hear and we talk more about threatening by external governments or by artificial intelligence, and we need to be aware of everything that is shared and everything that is actually open. How do you see and how do you are thinking right now about the right balance between open science and also in the same time being very careful about what we share? Don't you think, for example, that we should have some ethical committees to make sure of how those data are shared and what at the end of the pipeline, how they are used and in which hands they end?
I mean, I can just say from our perspective, on the policy end of the research that we're doing, the utilization of our platform as a sort of a database of free information to be used by any number of parties is something that, like I touched upon that, right? Majority of our downloads and our views are of our preprints. That's for some technical reasons, but also just because of what people are looking for when they're looking for scholarship. But we are more increasingly sort of understanding that our data can be used in other ways. The data that we publish on our platform, and the tricky part for us is thinking about how that translates to our relationship with our users and researchers. So we're coming at it from like a slightly different angle because we're making it easy to put data in a searchable and findable way and indexed way out into the world, and we're making it very easy. So there's a problem there, but that's also a part of our mission. So I think roundabout way of just saying we're working on it, and it's but we have to work hand in hand with research organizations and institutions to understand kind of what their needs are. And really just meet them. So we're kind of behind the scenes more.
Because some of your clients were business, law. It's not only research, right? In your Instagram.
So do people who use the platform sign some kind of agreement to, like, ethical agreement or something like that?
Yeah, we have as any software platform does, we have a terms of service with our users. So there are often violations of those terms of service. Users need to own the material that they put on the platform, but there's that issue, but there's also the issue of like who is utilizing the data that we have freely accessible on the platform, or that any researcher would put in any open repository for access. And I think the licensing issue around the utilization of data for artificial intelligence as training sets is a whole other can of worms that I can't really speak to, but maybe you can speak to it. I don't know. But it's definitely on the radar of the types of like repositories that the OSF is.
I think like data dissemination and data storage are like our two biggest problems. Like for us, we treat data kind of in different tiers. So like when we put, let's say, a study out and it's complete, then it's curated, that data can get put in the preprint in a journal, and we're okay for it to go out into the world. I think where our security concerns then come in is often around the patient data. So when we're giving out data to the world, we have different tiers from low level to mid level to like really high level restricted like patient data in terms of could you de-identify a patient. And so when it comes to CBIG, the requests that come in, they do have levels of security around the patient. It's ring-walled and laws of who can access it. When it comes to our type of data, like in terms of how does a cell line perform as a neuron, we can, once it's curated and it's, you know, past the QC and it all looks good to go out in the open, we're okay to put it out in any kind of accessible format for people to then access. I think then, in terms of putting it out there, that will be the kind of concerns of security, but then in terms of how we store it, curate it for the future, we have brought on a data manager to kind of address some of those kind of concerns. It's, you know, like if we have microscope images, how do you then store it over the next 5, 10, 15 years so that, you know, it's available and it's not going to get hacked and all of that stuff. So as platforms, I think it's going to become a bigger and bigger issue in terms of the data gets bigger and how we then manage it and store it, not just openly, but just if you're not an open platform, just as a closed platform, how do you manage that too? So Gabriel's the data manager, so he's going to give us the real answer.
Manage all the data. Yeah, well, I think Tom touched on the most important point, meaning that there's no sensitive data that's directly available out there for an AI algorithm to mine. Well, I mean, not that's released from the Neuro. So yes, so if it's deemed sensitive, then it goes through a data access committee, as you mentioned. Then there's the less sensitive data, which can get onto completely open platforms like OSF and others. And then I guess that's more tricky. There's often conditions that you can tie with the data sets, like the data person that accesses the data will, you know, okay to a condition like this data will only be accessed for health-related research or neuroscience-related research or something like that. And then if the AI algorithms comes and mines that despite this condition, I mean, there's not much we can do there, except that there will eventually be more regulation around AI mining algorithms, like it's currently ongoing for Midjourney and things that grasps all the internet for images and pictures without copyright consideration and uses that. So I guess it goes on all the same really complicated legal battles that are unfolding now. And then the next important thing is about consent. So these data that end up on these fully open platforms are fully consented. So I guess that's kind of theoretically fine. So the participant signed off knowing that the data will be made available online to everyone, essentially, or something like that. But then I'm not sure, maybe the consent needs to be changed, the language needs to be changed now, because informed consent, you know, implies that they are informed about what can happen with these data. And often in the consent forms you find today, it won't be listed something like, you know, your data will be available online, potentially subject to be mined by AI algorithms, which will build a chatbot with it, you know. But it might be important to add, I don't know. Yeah.
So I think we're out of time for the panel, so I'll keep my question to a comment, and maybe we can talk about it over the coffee break. But I wanted to come back to the publishing part, because I think a big part of the problem with top journals and publishing fees and everything else is that that's how research is evaluated. So my question would be, is there anything any of our panelists or anyone here could maybe talk about over the break, but are there things that we can do to change the culture around that, you know, if you publicly share a data set and it's used 1,000 times, is that worth more than one Nature paper that doesn't get cited? And I think that's where we kind of need to start changing how we think about sharing the science, but then bringing it back to how is the research evaluated, how is the researcher evaluated, how is the research team evaluated, and all those sort of contributions that go in that I know in this community, we often, our contributions often are overlooked, even though they're really important. So I won't maybe ask for an answer just because I know we're out of time, but maybe just something to think about.
I know that Tom has to leave, so. But yeah, this is a good discussion point for coffee or for dinner tonight for those who are coming. So the panelists should be there tonight for further discussion. So thanks very much, you three, and we'll see you in the next this afternoon and tomorrow. Oh, sorry, don't leave just yet. There's the tech talk, sorry.
I'm sorry to keep you away from coffee. All right, so I'm Gabriel Lapointe. I'm the application specialist for Evident, formerly known as Olympus in Montreal. I'm here to talk about the newest confocal we just launched on Halloween Day this year, the FE4000, and more specifically about its new detector. We are moving away from the classic multi-alkali and GASP detectors. The reason can be pretty obvious. It has a much higher photon detection efficiency. It has a broader spectral response, so up to 900 nanometers. But also, I don't know if you knew, but gas PMTs are built by hand. So there's a big variability between each of them. So it's becoming very hard to compare data from one institution to the next, or even if you have to replace your detector. The other thing is that unlike GASP, the silicon photomultiplier we're using do not decay over time, and they do not decay with exposure to light. So anyone who uses a multi-photon here probably knows what it can do. The SIPM, the way it works, the photon will give a response that is proportional to the amount of photon you get. So the peak will be twice as high if you have two photons hitting the detector at the same time. Its drawback is that it has a very slow response time. So it keeps sending a little bit of voltage and then it slowly decays. What that creates is this kind of convolution where you have multiple peaks close to one another. What happens is it kind of creates this ramping up that you can, at that point, it becomes hard to quantify. The way we work around that, we created a microchips that do the inverse calculation from that response. And at the end, we're getting just the peak and getting rid of that decay profile. What this means is that we're getting a very linear response. So from 1 to 2,000 photons at the same time. What does it mean in your imaging? A much lower noise level. Okay, when I say much lower, with about 2% power from a standard 20 milliwatt OBIS laser, you can use a resonance scanner at full speed, one pass, no averaging, no accumulation, and you will have problem detecting, seeing the difference between the Galvo and the resonance scanner. Okay. This image was taken at 0.01% laser power. Another thing that's interesting, if you compare the classic histogram to the one we're getting now, is that all of these small peaks actually represents the number of photons that hit the detectors. Okay? What does it mean is that this, the FlowView 4000, is always in photon counting mode. Okay, you don't have to think about it. Your student can do the experiment today, and 6 months from now, hey, we want to quantify it with actual photons, not random arbitrary unit. No problem, the data is there. Okay? A couple of years ago, came up with a way to count 2 photons that hit the detector simultaneously. And it was something big. Well, we just multiply that by 1,000, and we can do 2,000 simultaneous photon. Okay, so going from 1 to 2,000, impossible or almost impossible to saturate the detectors. Very low noise to begin with, so we got rid of the offset, we got rid of the gain. You only have laser power and dwell time, which is the exposure time. So basically that confocal is as easy to use as a standard camera-based microscope. Of course, new confocal is not just a detector. We can now go up to 10 lasers simultaneously, standard on the platform, including the 730 and 785 laser line. So for all of you working with tissues, iPSCs, it's much easier. Built-in laser power monitor with automatic compensation. So standard laser power all the time, comparable data. More channels, broader spectral response, better scanner, AI, noise reduction, and damn, 5 minutes is too short. I'll be available if you have questions. I'm here today and tomorrow.
So he's going to be here during the break, and maybe we break until 4:10 so that we have enough time to discuss and have a true coffee break. Thank you.
squeeze to talk, any questions? Yeah, I can see that. And then we're going to do the award for
I didn't mean to keep you here.
Yes. What did you quit?
Why?
Okay, fair enough. So do we have a laser? En fait, on s'est jamais vu en personne. Ah, ça fait plaisir. Tu te présentes juste après moi? Ah, super, super. All right. All right. Ce soir, je vais pas au resto, mais je suis désolé aussi. Puis, écoute, là, peut-être que Claire peut transférer les nouvelles, mais on a d'argent pour Roméo. On s'est mis dans un budget. Et le budget commence. Ouais. 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 Mais on se sent enthousiaste. Non, exactement, exactement. Puis à cette idée-là, il est lié dans les rêves. Yeah, I was looking for the name. I guess it wasn't that one. Ah, cool. Oh, okay, no problem. Absolutely. What I was looking for in that series of articles that they have is there one that summarizes all the time? Yeah? Okay, that's the one I was looking for. Like how many times could you reproduce the data? How many times the problem was, I don't know, the raw data was. Okay, because I was really looking for that one. Just a summary. Okay, so there's one. Okay. And I think she showed it, the COS person. Yeah, make that data. Yeah. Yep. Yeah. Yeah, there's. Yeah, right. Yeah. It was 86%. That was in the social one. Yeah. Yeah. Yeah, that's not 86%. It was popular.
get started again, my name is Jeff Ledoux. I work at UBC in the Center for Brain Health. Sorry. Okay, is that better? Okay. So I was saying I'm Jeff Ledoux. I work at UBC in the Center for Brain Health, and I've been helping out with the CNSP meeting. So we're going to continue on with our theme of open science. We've got 3 talks coming up about different open science resources from Carl, Judith, and Adrian. And we're also going to have the presentations from our CNSP award winners. So Brooke and New will present. Before we get into the talks, though, we're going to try again with the land acknowledgement that we didn't get to work this morning. So just let me try that. Okay, great. That worked. Okay, so our first speaker for this session is Carl Laflamme. He's the Structural Genomics Consortium scientist here at the Neuro, and he's going to tell us about the Icarus Antibody Characterization Project. So, Carl?
So I'd like first to thank the organizers, Claire Brown, for giving us the opportunity to present our work today. So it's a pleasure to introduce you to our open science technology platform that aims at characterizing commercial antibodies for every human proteins. So we're a group of 8 scientists called the Antibody Characterization Group, and we are an integral part of the laboratory of Peter McPherson here at the Neuro. We work in close collaboration with a public good Canadian company called Icarus, with the Structural Genomics Consortium, with key leading antibody manufacturers, as well as knockout cell line suppliers. Okay, so commercial antibodies are essential research tools used widely by molecular and cellular biologists to isolate and characterize their protein of interest. The problem, they don't perform as advertised by the manufacturers. So we've characterized maybe not over, but roughly 1,000 antibodies. And what we've seen is that a minority of antibodies are truly selective for the intended proteins. Whereas what we found is the majority actually either interacts with the intended proteins or unwanted proteins, or even worse, actually only detects unwanted proteins. And alarmingly, these underperforming antibodies here are often top-sided antibodies. So here's a global picture of the antibody liability crisis. So there are roughly 6 million commercial antibodies over there from over 200 suppliers. And most of them are actually not unique. They're cross-licensed between manufacturers. What's interesting is if you plot the 6 million commercial antibodies over the 20,000 human proteins, you'll find that roughly 95% of the human proteome is already covered by at least one antibody, however, with absolutely unknown quality. It is estimated that there's around $2 billion of research fund wasted yearly on the purchase of bad antibodies. And this has been a problem for over 30 years. So our approach to address this crisis is to use industry and academic endorsed protocols to screen side by side commercial antibodies and publish the data as quickly and as openly as possible. So here's our approach. So granting agencies nominate or fund the different antibody characterization studies. Then we work with a company called Icarus, who has developed partnerships with leading antibody manufacturers listed here and knockout cell line suppliers. These companies here provide us, our lab, with the different reagents. We pay zero. They ship the antibodies, and then we test them using the knockout cell lines, and we test them for Western blot, immunoprecipitation, and immunofluorescence. Then we consolidate the data into a report, so one report per protein, that we first publish on Zenodo, an open repository, and we're slowly converting these Zenodo reports, or these preprints if you want, into bona fide peer-reviewed articles at F1000 so that they'll be indexed on PubMed. We're also using the RRIDs, the unique identifiers. We use them in our reports, and we can actually use RIDs to link our data on the SciCrunch portal. So there's kind of three ways to access our data. So here's another view with more details about our platform. So targets get nominated by funders. Then we looked at protein expression to identify cancer cell lines with adequate protein expression. Then we request commercial cell lines if they are in the right cell type, or we made them ourselves. And for some knockouts, we actually work with the EDDU from Tom Durkin that presented before. Then we request the antibodies from the different manufacturers. We often get roughly 10 antibodies per proteins from roughly 5 different manufacturers. Then we first screen the antibodies for Western blot. And the idea here is usually we identify at least one specific antibody, and this will allow us to validate the knockout line, because you'll be surprised that roughly 40% of commercial lines are actually not KO. C-terminal antibodies often pick up truncated proteins. Then we screen the antibodies for immunoprecipitation and immunofluorescence. We prepare the reports, and we first share with the Icarus Advisory Committee, where there's one senior representative from each of these companies. We show the data, we discuss, sometimes they have questions, why is my antibody not performing well, blah, blah. Once we reach our scientific bar, then we're ready to publish on Zenodo, and then on F1000, and so on. So here's an example. So I'm gonna show you Western blot IP-IF data for this particular protein, TMEM106B. It's the same for all 85-ish proteins that we've done. TMEM106B was actually nominated by the Michael J. Fox Foundation. TMEM106B was found quite recently in brain inclusions from patients suffering from various neurodegenerative diseases. Parkinson's is included. Okay, so here's the Western blot data. So for TMEM106B, we got 1, 2, 3, 4, 5, 6 different antibodies from, I think, 4 manufacturers. Each blot corresponds to one antibody. The name of the antibody is above each bar. When there's no star next to the antibodies, it means it's a polyclonal antibody. And as you know, polyclonal antibody suffers from batch-to-batch variation. These are not renewable reagents. When there's one star this antibody comes from a mouse monoclonal hybridoma, they are considered renewable. And 2 stars is really the best standards. These are recombinant antibodies derived in vitro from expressing plasmids. And okay, what do we see here? So this antibody here gives one band, that band disappears in the KO. So this antibody recognizes TMEM106B. The manufacturer do not recommend this antibody for Western blot, but it works really well. This antibody do not pick up TMEM106B. However, it is recommended by the manufacturer. This antibody is great. It's recommended by the manufacturer. Cool. This one is the top-sided one. Personally, I wouldn't use that antibody for Western blot. This antibody is recommended by the manufacturer. However, it's absolutely not suitable. And this one actually works, but it's not recommended by the manufacturer. All right.
next, we screen the antibodies by immunoprecipitation. And there's various ways to evaluate antibodies' performance for IP. We could have done an IP mass spec, etc. Here, what we do is that we do an IP, and we compare the amount of TMEM that is captured compared to the starting material using a Western blot, which we use an antibody that was validated for Western blot just before. So a high-quality antibody for IP would be something like this, where you have an enrichment of the target in the IP fraction compared to the starting material. And sometimes the antibody even depletes the target protein from the unbound fraction. Actually, most of the antibodies perform well by IP. However, mostly were not recommended for this application. So our platform also is able to identify new application for certain antibodies. And then we screen the antibodies for immunofluorescence. And to be honest, this is by far the most challenging application. To avoid antibody user biases and imaging biases, we use a mosaic trick where we label the wild-type cells with a fluorescent dye, the knockout cells with a different fluorescent dye, which allows us to combine the cells the same well, and we can differentiate them because we pre-labeled them with a dye, and then we do the antibody staining. So we always show the DAPI staining, and then with Joel at the ABIF, he was able to develop a code that can segmentate the wild-type cells here in green and the knockout cells here in pink. And we can also make performance ratio between wild-type cells and knockout cells. It's already cool. So this antibody here is recommended by the manufacturer. Is it specific? No, it's not at all. It's the same signal between the wild-type and the knockout cell. This antibody here is also recommended for IF. It's crap. However, this antibody here that is not recommended for IF is well performing. Okay, so this is the kind of reports that we publish. These are guides that researchers can use to select the most promising antibodies for their research need. It does happen where we screen all commercial antibodies and there's not a single one that is specific for the intended protein. So we work with a willing consortium of antibody manufacturers that agrees to generate antibodies for our target list. Here's an example for SMUC1. So SMUC1 was found by a consortium working on Alzheimer's disease in the US as a potential biomarker. It goes up in 80 patients. However, the antibodies were terrible and were all polyclonal. There was not a single renewable antibodies that was working well. So Abcam raised his hand and said, hey, I'm gonna make some good antibodies for you guys. And indeed, it worked beautifully. And the team that nominated this protein had access to these antibodies prior to the release of these antibodies on the market. So in conclusion, from characterizing hundreds of antibodies, we're now able to quantify the antibody liability crisis. What's the real picture? We believe we have an efficient strategy toward covering the human proteome. The very good news is now, today, without doing anything, 50% of the human proteome is already covered by a high-quality renewable antibody. And this is based on extrapolation of our data to the human proteome. And we really believe that the first thing we should do is to characterize the already available antibodies. And when there's a gap in the market, then we can focus on generating recombinant antibodies. Because at the moment, there's already so many companies generating so many antibodies for proteins that are probably already well covered. So let's characterize the already available antibodies and find some good ones. Something that I would like to say is the antibody characterization group is part of a bigger picture, of a bigger family. So we're under the umbrella of the Structural Genomics Consortium, and the SGC's mission is to accelerate drug development. And they do this by following strict open science principles and by generating high-quality protein reagents. They have 6 sites worldwide with their own unique expertise, and all SGC scientists contribute to the development of target enabling packages or TAPs. So you would have one TAPs per protein, and with a TAP, you would have a high quality protein structure, high quality protein probes, selective antibodies, and so on. We're also working together with the Only Good Antibodies community that led from Leicester's University, who are more downstream of us, who are working with different antibody stakeholders such as publishers and grading agencies to promote the use of only good antibodies. We're working with PsyCrunch, with ICRIS, its partners, with the IBIF. Slowly we'll be implementing Omero in our platform, and Dr. Lacoste will talk to us about the Omero just after me. And we're also working with the Early Drug Discovery Unit. So these are our different partners. I'd like to thank everybody, and I'll take any questions. Thank you very much.
So I'll invite Judith to come down. While she's getting set up on her presentation, we do have time for a question if someone has one.
I'll ask you guys a question. When antibodies are recommended for immunofluorescence, in your opinion, how much actually failed for that application when tested over knockout cell lines? And those that believes that, 80% of antibodies recommended for IF actually works. Maybe you can raise your hand. And what about 50%? Ah, not too bad. It's actually 40%. All right, thank you very much.
Thank you. Okay, so our next presentation is from Judith Lacoste, who's the founding president and scientific director of her company called Mia C'est la Vie. And she's going to tell us about some resources that she's set up for data storage and data sharing.
How do we make this full screen? Okay, thank you. Forgive me, I'm not a Windows or Mac. I'm a open source Linux person now. But sometimes we suffer. All right, so thank you everybody for being here today, and thank you to the organizer for allowing me to present my work, which was done under the umbrella of Canada Bioimaging. So, where's the cursor? Okay, so we already had a nice land acknowledgement slide, so I'm gonna skip this one, which is not as nicely done. Okay, so we are interested in fostering automated, open, reproducible, and fair science in line with the bioimaging communities and standard. For confocal microscopy, the International Standard Organization released a confocal ISO standard back in 2019. And there are two organizations that are very active in implementing this confocal ISO standard. Here in North America, we have Bioimaging North America, and Canada Bioimaging is working under the umbrella of Bioimaging North America. And around the world, there is the QUAREP-LIMI organization. QUAREP stands for Quality Assessment and Reproducibility for Images and Instrument of Light Microscopy. So there are several of us that are very active in these organizations. And it turns out that we were co-authors on the Nature Method issue focusing on microscopy reporting and reproducibility. So automation means that we need to get information that is both human and machine readable. Open refers to accessing your data mostly at time of publication, but possibly before, depending on the context. In terms of reproducibility, being an author on the Cancer Reproducibility Project at the Center for Open Science, we need to consider the performance of confocal microscopes and quality control metadata that results out of it. We want to have findable data that we can search using all sorts of queries. We need to make sure that the data is accessible through authentication and authorization. We want to make sure that the data sets we generate are interoperable between different technologies we're using, whether it's genomics, flow cytometry, or microscopy. And we want to be able to reuse our data set and potentially reanalyze them with new hypotheses or new analysis tools. Okay, so in order to achieve that, we need to seriously consider research data management policies and organization. So we need to share and get organized for both data and metadata. And metadata is data about data, right? So it becomes a really blurry. So if you want to have a very defined definition, it does not exist. It gets blurry very quickly. But the whole idea is that before publication, it's about organizing your data so that you can find yourself into all the data that you have. Obviously, putting in place good storage strategies. And at the time after publication, we need to consider how we're going to share and preserve the data set for the future. So what I want to, this thing keeps going higher than my height. Okay. So what we have here on the right is the recommended metadata for bioimaging model. We call it the REMB. And this can be applied to any type of modality, but we're focusing here on data that is generated from microscopy. So you can see that you have the microscope here, but before we get on the microscope and after we get images, there's a lot of information that needs to be collected and curated if we want to achieve fair data. So here in Canada, we have research data management policies in place, and this is stating that grant recipients are required to deposit into a digital repository all the digital research data, metadata, and code that directly support the conclusions that are put out at the time of publication. Okay, so we're lucky because despite that, the Canadian government did put together an infrastructure that is going to help Canadian scientists to become compliant with the research data management policies. This is what we call the Digital Research Alliance of Canada. So the mission of the alliance is threefold. The first one is to provide storage capacity to enable research data management. Secondly, it provides advanced research computing clusters for data processing and analysis. And lastly, it supports research software development. So as you know, Canada is a very large country, and the data centers are spread out across the country, and they are linked to one another with high-speed internet infinity band connection. So this resource is available to Canadian scientists. And the way, at the level of data analysis, how this can be summarized is the following way. You can have your data stored on the Alliance storage place. And when you work out a data analysis workflow, you submit this workflow by logging into the Alliance logging node. And then you're going to submit your job to the scheduler that is instructing the computing clusters how to proceed with your analysis workflow, where to get the raw data file, where to save your output, and so on. So the scheduler will then dispatch the job to the computing clusters node where your data processing and analysis will be executed. At the end, the output of the workflow can be sent back to storage and linked with the original data sets that were analyzed. In the province of Quebec, we have 2 data centers here in Montreal at the École de Technologie Supérieure and one at McGill University. And these 2 data centers are maintained and supported by the Calcul Québec organization. And Calcul Québec, or Compute Québec, offers a number of trainings for scientists to get familiar with in how to use the structure of the alliance. It offers a number of introductory classes on how to use the Unix command line system, version control with Git, virtual environments using Jupyter Lab, and a number of resources that are based on the Python language. So I know that not all of you are microscopists, so I'm going to try to go through this very rapidly. But what is peculiar with microscopy data is that just having the raw file in your hand is not going to be that useful. And the reason for this is that the information that is contained in those files need to be presented in an array. So what we are looking at right now is a microscope, a microscopy image generated from a kidney glomerulus. And the background is dark, and the feature of interest is showing up as light, bright gray and white. Now, if we enlarge this tiny section here, you're going to see why we have pixels, right? So these are picture elements. And when you blow this up, now you have just a 1.5 micron area that is showing you the positive signal, which contains a lot of gray level information, and the background information, which contains a lot less levels of gray. So if you just have a list of these XY coordinate with the intensity level for each one of them, it's not very informative. So in microscopy, it's important that we see the picture before we actually do anything else for data analysis. So this information is usually collected as gray levels, but we can apply a false color, and in this case, we were looking at lipids that are contained in this kidney glomerulus. But most of the people doing microscopy will actually repeat the staining with other markers, so that in the end, the image contains information from different components, and the images are overlaid with one another. So really, a microscopy data set has several dimensions. When we are dealing with very thick specimen, we have the ability to generate images in the Z dimension, and when we reconstruct these images, we now can generate 3D projections like this, which we can zoom in, zoom out, and spin around pretty much like you would do with Google Earth and Google Map. So the take-home message is that microscopy data is multi-dimensional, X, Y, Z, color or component or channel. And if we do this on a living specimen, there's also going to be a time component. So they tend to be large data sets in the terabyte and petabyte. And what is really peculiar is that before proceeding to data analysis, we usually have to do a visualization step in order to really decide what we're going to focus the analysis on. So this information is also associated with an important amount of metadata, and for example, for just these images, this blurb here is actually running for several pages. So this metadata is heavy, but it's actually essential for further downstream processing analysis and interpreting the outcome of your experiment. So we need a lot of computing power and speed for visualization and for data processing and analysis. Now, we are lucky in the microscopy world because we have access to an open source software called Neuro. Neuro was developed by an organization named Open Microscopy Environment. And Neuro can be envisioned as a database, but it does a lot more. So as I said earlier, the visualization component of Neuro is extremely powerful. It allows people to peruse their data set and look at all the dimensions, X, Y, Z, time, and color that are present in the microscopy data set. The other very powerful feature of Neuro is that it has the ability to read the 160 different file format that we have in microscopy, so you don't have to worry about converting between file format all the time. Now, Neuro, provided that you properly annotate your data with tags, with key value pairs, and so on, it becomes a very powerful tool for data management, and you can tag and query and retrieve the information you're looking for. But it's also very powerful in the sense that it links with a whole bunch of quantitative analysis tools that it becomes very useful down the road. Okay, so because Neuro is an open source software, the OME organization has put out a lot of information for people to go ahead and install their own instance of Neuro in their lab or in their institution. And here in Canada Bioimaging, we have Steve Ogg here, who is at the time was based at University of Alberta. And Steve went ahead and read the documentation for installing Neuro, and he also went through the documentation offered by the Digital Research Alliance, and he was able to create for us a cloud-based Neuro instance that 2 minutes. Okay. So, so we have this cloud-based instance, and we were able to proceed and come up with ways to automate the process of uploading the data sets as well as the metadata into the database. So essentially, we have a lot of metadata for microscopy, and I'm not going to have time to go through all of that. But there's a link here to provide more details. But the bottom line is, at the end of the day, when we automate to this process, we have captured a good amount of the metadata in an automated manner, and we can still associate metadata. Okay, we can still associate the metadata coming from the analysis workflows and have this linked into the Omero database. So I want to finish very quickly by saying that there are a lot of us struggling out there to properly automate the annotation process for microscopy, and the BINA organization is putting together a number of people that can share both solution and challenges, and the idea is to work together as a community and come up with a way to gain better control over research data management so we can do open, reproducible, and fair science. Thank you.
Maybe we can do a quick question while Adrian's coming down.
Do you have any collaboration or with microscope manufacturers or the software developers to standardize the metadata?
Oh yes. So the QUAREP organization is an international organization. There are 500 members, and we have the microscopy vendors that are actively working with us to establish the metadata model, and it needs to be revised because we need to go into the next generation file format. So yes, the manufacturers are working with us to get organized with this and streamline the process. Thank you.
Okay, so our next speaker is Adrian Peyrache, who is an associate professor here at the Neuro, and he's going to tell us about his efforts in open systems neuroscience.
Good afternoon, everyone. So I usually work on sleep in the lab, but I will change subject today. I don't think it's the right time for that. And I'm gonna tell you a bit about what we're doing on open system neuroscience, as I like to say, in the lab. And for us, it's been actually quite a journey because yes, we're a research lab, and we started to develop like many tools for the community and for open science without really any, training or knowledge of exactly how to organize this. So I'm gonna share with you a bit of of these adventures. Like, how do I change slide? Is there a keyboard somewhere? Here? Yep. All right. So system neuroscience is basically this discipline of neuroscience in which we try, when we say we because we're fully part of this, to record basically, as many neurons as possible at the same time in living animals and relate it to cognition. It could go like to sensory perception, memory, motor planning, you name it. But it's really about like decoding what is going on in the brain and trying to understand like how neurons communicate with each other to actually, make decision or process all these cognitive abilities. And here you see actually on the left some of the most recent development, for example, in the field of electrophysiology, meaning like recording brain waves and the activity of single neurons in the brain. This is one of these electrodes called the neuropixel. It's about like 1,000 channels recorded simultaneously at pretty high sampling rate. And you end up with like this huge data set from neuronal activity in the brain. You have also other possibilities to do that with optical imaging, and recently up to 1 million neurons were simultaneously recorded in a living animal. So I just let you imagine how much data that's represent, you know? And not only it poses great challenges to store the data, but also to process and share the data with the community, all right? So with all these challenges in mind, actually it's good to look at what is happening in the other field, because neuroscience is not the only field in science that generates and process like large data sets. Astronomy was actually perhaps one of the first. And there is actually a very interesting case here. It's how in the early 80s, the field of astronomy started to think about like to come up with a common data format to share data in the community. And they come up with this idea of FITS, for flexible image transport system, and it's still used today by everyone in the field. It's available in all possible coding language. You have wrappers for C, Python, you know, whatever. And it's readable by like most softwares, including, you know, ImageJ, Photoshop, GIMP. So it's really something very common. And there is a funny story here is that the European Union launched a new satellite about 10 years ago, and they wanted to develop a new file format for that. They spent about 10 to 15 million euros for it. And the first thing the community did was to actually write wrappers to, you know, transform the data format into FITS. So it was a total failure. And you see that there is some kind of, you know, once a format is available, people tend to stick to it. And in astronomy, it also makes a lot of sense because, like, it's really, they have this dichotomy in the field between the data providers, right? You have like people running these large telescopes and collecting a lot of data, and data users. And usually these people are not necessarily the same. Data users, or perhaps you're gonna spend 6 months in Chile, you know, collecting data. Then you go back to your lab and you analyze the data for 3 years. And this separation is very clear, and you know, it really required for them in this community to talk to each other. Problem is that we're not astronomers in neuroscience, not yet at least. And the reason why I'm saying this here is because the data we collect are of extremely different types and different modalities. Here I'm just showing 2 examples actually from our lab. On the left you have electrophysiology, so this beautiful electrical wave generated by the brain at many different time scale, from like not even a millisecond, the activity of a single neuron, to large wave that, you know, entrain hundreds of thousands of neurons, which can be as slow as like, you know, only 1 cycle for 10 seconds. And on the right side here, you see calcium imaging, right? So now you have these portable cameras on the head of a mouse, and you image, like literally imaging the activity of neurons that, like, we use calcium indicators as a proxy of neuronal activity. So totally different modalities. On top of that, we're doing behavior, and behavior can be many different things if you have like a typical system for that, but it could be, you know, like decision, position of the limbs and the whiskers, the eyes, all sort of different modalities that you have to accommodate and incorporate in your data and in your data analysis. All right. So how do we handle all this data together? So like years ago now, 2014 about, we had this symposium at the Howard Hughes Medical Institute, Janalia Farms, where a lot, you know, of people, and I was lucky enough to be part of them. We sit down for 3 days in a room and started to think about, like, okay, what do we do now with all this data? And we came up with the idea with this file format that was called Neuro Data Without Borders. So it's a very flexible data format that can include data from many different modalities, behavior, also all the information you need from individual animals, so that everything is properly documented. Anyone, and the idea is that anyone should be able to download these data files, open it without knowing anything about the experiment, without talking with the data providers, and be able to access immediately and very rapidly all the information this person, the data user, needs to analyze this data. And the development continued over the years. More recently, the version 2 of the file format was released, and really here you have a quick, overview of the kind of data that can be incorporated. And also it's flexible because you can add, of course, custom data to the files. So it's growing. It's been supported by many different organizations worldwide. So it's getting there, and at least it's one possible solution. All right. However, there's still a very big challenge here. Is that yes, we do have a solution to share data, to store data, but it's complicated, right? Because how do we actually now analyze this data? And for that, it requires a common language. All right? It's not enough to know how to store the data. You need also to make sure that you can analyze it. We want everyone to be able to use it, to use the data. It has to be fully open source, ideally relying on Python or other open source programming language, not necessarily MATLAB, which is close, not totally open source. And we want communities to develop high-level tools, softwares, based on this common framework to analyze, data for data users down the line. All right. So we worked a lot in the lab to develop like this framework, like a language to analyze data. And at some point I was like, okay, we've done a lot of work. It will be great to actually release it publicly. This toolbox that we had in the lab, we called it Pineapple for Python, analysis package, and it meant to be like very light source with very few dependencies, so that it is easy to maintain, stable in the long term. That's still actually a key aspect of this toolbox. And we put the package together, put it on GitHub, do a public release, and here you see the result of the tweeted, the X, now we call it, posted at the time, and it's relatively speaking, got somehow viral, like a lot of different of likes and retweets, so we were super happy about it, and people started to actually contact us to contribute and to help us. So it's been doing great since then. It's growing, and let me give you a very quick tour in just a few minutes without getting into the details of the code, of course. Just also on a quick note, the whole package, the first version of this just got described in a paper that was recently published in Elite Life. So, choice of architecture. When you come up with a language framework like this, you need to think about like how data are going to be organized and how you're going to basically put together this framework. And we use a method in programming called object-oriented programming, meaning that data are part of it's an object that has its own method, its own description, and that can inherit also the method and properties from other objects. And it's really interesting, without getting into the details of, again, programming here, is that you break down a complex problem into smaller problems, all right? And you can reuse the code of other very quickly, very efficiently, and it's robust, because once the object is created, it has its own definition of time, its own definition of values, so that it's not just a vector of value that is in your memory workspace. It is an object that is really robust against any kind of misusage of the data. So it's very standard in the field, but just for you to share, there was other possibilities here, but this is the choice we made. Here with just showing you the kind of thing you can do. These are the 4 totally different experiments, like slice recording on the top, where you're basically gonna inject current and use a drug and wash it, or electrophysiology in freely moving animals, or here like in a head-fixed animal, or even in a non-human primates, recording neurons. And the idea is that basically all these very, very different data can be compressed into a very small number of objects that will describe the data, all right? And then basically use methods that are pretty, intuitive to manipulate this data in different ways. You can restrict, you can align the data, you can do all sort of very kind of primitive function, so that at the end you generate the kind of figures that you see in papers in the field, which are like high-level kind of analysis. To this end, actually, behind the scene, all the operation you can make are the following, right? So you see that they have the same name, so you have the different objects here in columns and the different methods, and you see that actually it's about like doing some very primitive things, is that you're gonna restrict some events to an epoch, all right? You're gonna count the number of events you have in Windows. You're gonna align the data with each other. And this is the kind of thing we do over and over in our field with this very large data set to align some, for example, behavior and neuronal activity to synchronize the different data stream and so on. You can do the same also like epochs in time. They have like different ways of playing with them, combining, merging, or excluding some of them. I'm gonna skip this one for the sake of time. I'm just gonna give you like 3 examples to show you actually how easy it is almost to analyze data within this framework. So starting with raw data, which means like the time at which different neurons have emitted action potential, and a feature, behavioral feature, meaning the angle of the, in this case, the angle of the head of the animal as it's freely moving in the environment. You can, in few functions, actually at the end, pull out the figure in which you see the activity of this neuronal population color-coded as a function of the direction of the head, and you can even decode the feature from the neuronal population, meaning you can decode what is going on in the head of the animal in just 4 lines of code in this framework. You can do it also for other modalities. In this case, it's interesting. These are publicly available data set from the Allen Institute in Seattle, and actually, it's all based on NWB, which is, fully supported within the Pineapple framework, so meaning you can directly download data from the Allen Institute and other archive in this framework and analyze them. It's pretty cool. And do all sort of analysis here, for example, like which orientation of visual fields these neurons are most responsive to. And here, like even data collected in human epileptic patients, also publicly available, and do some kind of analysis cell by cell to see how neurons here are modulated by these different images. All right? So this is a big framework we're building. We started with this core package for analyzing data, but now we're expanding this in different direction. So we have this cocktail of many libraries. We call it Pina Colada that we're working on, and different things to actually make it a better tool. So it all started with, sorry, I don't remember. Yes, sorry. Code is fully open source. Anyone can contribute. Of course, it's all on GitHub. So like pretty classic, and we're building a community right now. We started just to actually have workshops. So the first one took place just a 3 weeks ago at McGill for McGill grad student, but now we're planning workshop for the community in general and doing it not only here in Montreal, but elsewhere. So it all started just by some work in the lab. We actually had a grant. I realized actually before that I forgot my thank you slide, so I'm just doing it orally. So we got money from TOSI to actually make sure that this workshop can happen. That actually I can pay the postdoc also who's not doing science anymore to work on this project. So this is him, Guillaume, who's standing up over there. I don't have a better picture of him, I'm sorry. And actually got hired by the Simons Foundation in New York a few months ago to continue the development of this. So I was really happy and proud of him for that. So in conclusion, data storage and sharing in system neuroscience has made great progress with a common file format like Neurodata without border, NWB. It was, for us, we realized it was time to develop common analysis pipelines, so you can share the data, now you can very easily also share your code and make sure that everyone can run this code easily on their machine, which is too often not the case. And this will ensure reproducibility of analysis and community-driven efforts to develop open source tools. All right, thank you very much for your attention. I will take any questions.
Questions? Yes, there is. Please.
Hi. Thank you for your talk. Absolutely wonderful. But I come to the realization, perhaps because you talked about astrophysics before, that there is a common thread to all the discussions we're having. Managing data on a huge scale, but also generating data on huge scales. And regardless of the science we're talking about, we're generating them in a very short amount of time. But it takes months, if not years, to analyze them. So maybe this question is difficult and certainly philosophical, but we're looking at storing geometrically increasing amounts of data with no possibility to analyze them successfully before it keeps increasing. What's your take on that? I apologize for the difficult question.
No, it's a great point. So I think that, like, okay, so let's break down this question. In labs like mine, you know, every data set is collected by someone who will analyze it and publish at least one paper with this data set. Let's put it this way. And very likely, if the data set is of great quality, it will be used in other publications. In the lab right now, I do have people who are not running experiments anymore because they realized that they didn't really like, you know, spending their days with mice, but better with a computer. So they actually do their PhD just analyzing data of others, all right? My own data sets from my PhD, which early on I actually made it available, was, I think, used in about 10 to 20 other papers. So, you know, I'm happy about it. Then there are all these data sets that have been collected and may not be used that much. So if you take the Allen Institute, for example, now, they're collecting this gigantic data set, recording, you know, from tens and tens of mice. It's amazing what they're providing with the community. Whether every single data point collecting there will be used in papers and in journal, that I don't know, in articles. But at least these data are available, and we can hope that no one will actually do these experiments to collect similar data. And that's the whole point, right? It's not only about reproducing all these the results. It's also like make sure that we're not using living animals for nothing. And I think it's like top priority here. Yeah.
Thank you.
Okay, so I think we're going to move on to the award presentations. Let's thank Adrian one more time.
Thank you.
Okay, so we have 2 awards to give out for the CNSP. The first one is our 2023 Platform Administrator Award, and this is going to Brooke Ring. So Brooke, could you please come up and we'll do a quick photo.
Down. Okay. Hi. Thank you very much for letting me come here and speak today, and thank you for the award. I'm coming from Queen's University, where I am here to represent the Queen's Cardiopulmonary Unit. And as part of today, I'm just going to tell you a little bit about myself and the core facility that I run.
So, just a little bit about me first. I am a graduate from Trent University. I have a dual degree in biochemistry and molecular biology, and my master's in life sciences. I got my first opportunity working as a lab manager at Trent and then a course instructor before deciding to move on to human medicine, where I worked as a hematology research assistant. And then I worked doing that for 8 years. I then moved on to managing several labs in the pathology department at Queen's. And I got my first taste of being a core scientist working for the Queen's Lab for Molecular Pathology, which is a very small core. In 2019, I became the manager of the Queen's Cardiopulmonary Unit, and as of this year, I am the associate director of the Translational Institute of Medicine. I'll briefly go over my own personal research. So I used to study a gene called TET2 and its mechanism and impact in blood cancers, specifically myelodysplastic disorder, which is a precursor to AML. I created a genetic discovery panel for the diagnosis and early detection of blood cancer. And then in 2018, I worked with Thermo Fisher to create a commercial panel that ended up being used as a diagnostic panel in the clinical, and it's used all around the world. And finally, the platforms that I am familiar with, once upon a time, confocal microscopy, flow cytometry, multiplex biomarker analysis, so like nanostrings and Luminex platforms, but my specialty is in next generation sequencing. So now, to the bulk of the presentation about my core. So as I said, I work for the Queen's Cardiopulmonary Unit. This was a $10 million CFI founded by Dr. Stephen Archer. Dr. Archer is a renowned cardiologist and pulmonary hypertension specialist. And this core was originally founded by him to do research in blood, lung, heart, and vascular disease. But since we have made it an operational core, it has expanded widely to all sorts of different things. I'd like to highlight the 4 things that I think make my center unique. Oops, I skipped ahead. So we are a full-service core, which is that my scientists and my team not only run your samples and analyze the results, we help design the experiments, we help write grants, we collaborate, or we find you collaborations for publications. At the same time, we train students and staff on the techniques in the process. We help troubleshoot and refine the projects. And we want to ensure that your samples and your project is completed and maximized and elevated to the highest level. The next thing that makes us unique is that we're not just a microscopy core or a flow core, we're actually a whole bunch of different things. We have histology, CT scanning, NGS, as well as flow and microscopy, all in one building, all in one 8,000 square foot area. And we are a site for translational medicine. So Queen is known for its translational medicine program, and this is the idea behind creating a bridge of basic scientist and clinician science. We ensure that there is kind of a place and a pipeline that's for the basic scientists to get their discovery therapeutics to the other side into the hands of clinicians who can treat patients and do clinical trials with them. Queens is home to the Translational Institute of Medicine, for which I am an Associate Director of Operations, and one of the features of that is a Translational Medicine graduate program, which is a classic health sciences research program married with kind of medical school. So we have students that go into the clinics with physicians. QCP is well spaced under the direction of Time now because it actually has a Kingston General Hospital satellite clinic in it, which runs daily echoes and PFT, so pulmonary function testing, which has a really large research capacity and clinical trials capacity, as well as running just pediatric clinics for cardiology and respirology. So we have this ability to have blood taken in our clinic and analyzed through the other door. So it's a very unique site for that. And finally, our core is very much expertise driven, which you'll see in a second. And it is the thing that people come for. So yes, we have really fancy state-of-the-art technology in our facility, but it's the experts that actually run the equipment that people come for. So I'd like to highlight them now. So you already heard about me. I'd like to talk about my partner in crime, the other associate director of Time, Dr. Charlie Heimarch, who is here today. He's also the manager of the discovery cores in another department, and he is also our genomics, transcriptomics, and molecular medicine lead, where he runs a lab that has all the equipment in to do next generation sequencing and create libraries and quantification and PCR machines like that. The next one is Dr. Alahe Alizadeh, who runs our small animal imaging and preclinical testing facility. She runs an MI Labs Vector 4 CT scanner, which does CT, SPECT, PET scanning, and she also does radioisotope labeling. She's a great example of someone who has 3 degrees. She's a nuclear physicist, an engineer, and a medical imaging specialist. She is very unique, and I think there's only probably just her in the whole world. At least we like to think so. And if you're a cancer researcher that's just kind of getting an idea about, maybe I want to do some radioisotope labeling for some cancer cells, she will not only help you decide what the best, probe to use is, how the best technique to use. She will help you get all the permits in place that you need and the ethics, as well as run the samples for you and help you analyze it. So she's a really good example of like the full scope of full service that we like to encompass. Next is Dr. Patricia Lima, who some of you may know. She is our molecular imaging and cytometry specialist, and she runs a Sony SH flow cytometer and sorter, and she also runs a super resolution 2-photon intravital microscope, and she is really excellent. Next, we have histology as well. This is under Oliver Jones. He also does transmission electron microscopy. And this is he worked in both the clinic side as well as the research side. So not only can he just blast through a bunch of histology if he needs to for large projects, he can also take really weird customized projects where he can kind of work with the researcher to get the right orientation or do the right special staining. Which is really helpful. He has to do polymer work for the research department in chemistry, slicing blueberries and all sorts of weird things. If you have money and an idea, we'll try and help you. And then finally, we have Curtis Norhoff, who assists Patty in the molecular imaging and cytometry lab, and his specialization is cell culturing, proliferation assays, and the microbiome. So what is the impact of QCPU? So we opened in 2017. And we operationalized in 2019. So in 5 years, I think we've had a really positive impact at Queen's. We have 81 investigators and their labs that are users at our facility. 15% of that is engineering, actually, and 4% is external. We've had over 150 trainees, 85 publications, 600 citations, and we've played a very vital role in recruitment and retention. QCPU offers its hours of service in what we call super user packages, where you bundle up like 100 hours and sell it ahead of time. And these have been used as recruitment perks in the interest of recruiting the best and brightest. And it also has been highly impactful on early career researchers, especially over the pandemic, of getting them a foot in the door on their research when they couldn't hire students or they couldn't get here on time. Finally, QCPU has collectively worked with scientists to about $25 million worth of research funding have come into the university to do partnerships with our scientists who have either collaborated or co-PI'd on the grant. And this has helped the university immensely in growing the extramural funding envelopes that come to the university. And that is time. Thank you very much. Thank you for CNSP for the award.
So maybe a quick question for Brooke while New comes down and gets set up.
Oh, it's really high. I can hear you. So that's a really great question. One of the things that we do is that we have usually we have our director, Dr. Stephen Archer, champion a grant if it's not a faculty member, but one of our team members, Dr. Charlie Heimarch, is a faculty member, and because he does bioinformatics, we have a lot of flexibility in applying. But with university permission, any of our scientists can apply for a grant, as long as they have the VPR's support. Thank you. Any more questions? Great. Great job. Thank you.
Okay. Okay, so our second award winner is New True from University of New Brunswick for the 2023 Platform Scientist Award. So we're just going to do the quick photo again, and then she can tell you about her platform.
Let's see if I can fix this. Hey, everyone can hear me all right? Well, first of all, I really would like to thank you, the Canadian Network of Scientific Platforms, for the award. If I'm going to be terribly honest, I don't really think about this as an award for myself, but actually an award for my facility, because I think we're pretty awesome. I'm going to have to say that the last talk was a little bit of an act to follow, because we're on the opposite end of the spectrum. But I do think that there's still value in what it is that we do. So here's some lessons from history and collaboration. And the title is Not Too Cool for Old School, because our facility has quite the extensive history. So in case you guys ever decide to visit us in New Brunswick, we have a beautiful campus at the University of New Brunswick. We are located in Bailey Hall, so the biology building, pointed at the arrow. And I'm sharing this in case we ever do a conference in the area and you decide to come. So the facility was first started by Jerry Bantz. He started in the biology department in the 1960s. And in the 1980s, the facility became the electron microscopy unit because we needed to be a separate facility to do work for other departments. So instead of just doing stuff in the biology department, we was doing things for chemistry and engineering and so on. And it became the microscopy and microanalysis facility in the 2000s with the acquisition of a Leica SB2 confocal. So the MMF staff, on the left here is Douglas. So he's my boss. He's the director, and he's also the senior X-ray microanalyst. On the right is Stephen Cogswell. So he is a support engineer, but he also does all the chemistry and physics-related microscopy work. And for myself, I do all the biology stuff. So I do everything from histology to cryosectioning to SEM to TEM. And all the sample preparation. So when you think about microscopy, you sort of think about, we just work on the microscopes and researchers bring us our samples and we sort of call it a day. And sometimes we had to work with really old microscopes. So, so this is a JO733 microprobe. It was made in 1986. We received it in 1994 from the Research Productivity Council. And the lesson here is get the machine, force the hire. So this is why my boss was hired, because we needed someone to run the microprobe. And it's still going. How? So meet fish gill. So this was an extra microprobe that someone was selling. First, get yourself an engineer. No, you cannot have ours. But we were creative in finding parts in order to replace some of the parts of the machine, so that's why you see all these black panels intermixed with the brown. We also have a JO6400 SEM, acquired in 1990. It's still going. And the lesson for this one was make friends with the service people, because the service people, because we made friends with them, they gave us the secret documents about some of the diagrams of the electronics that we use to keep this machine going. So other machines that we have, since 2003, we had the Leica SP2 confocal, we had the JO 2011 TEM. Since 2008, we have the Hitachi SU70 SEM, but that was acquired in 2019 and still in another building. And since I've been working in 2016, we got in the Leica SP8 confocal, we have a SIOS 2 dual beam FIB SEM, and then histology and cryo-sectioning specialty that I brought in as part of my background. So a lot of microscopy stuff, but that's not everything that we do. There's more than one way to fix something. So this is a picture of a dual TM grid holder that students keep on unlatching and breaking the little brass bit there. And so our way of dealing with that was we put the samples in for you, right, to optimize working with students. We are also photographers plus. So back in the day, you actually had to take your microscopy images, and then you had to develop it on film, and then you had to expand it, and you had to put little stickers on it. Now we do MSJ, putting things in Photoshop to make bigger panels. We're also interior designers, so sometimes you need to put things into a new building, and not into a new building, you need to make space in your existing building, because someone's got in a machine, and now you got to make it fit. So here's us reworking this room so that we could fit the SP2 Leica. Code red, so dealing with trying to fix things. So you pull the back of the microscope and you deal with all of the cords, and sometimes you're like, okay, there's an air leak somewhere, let's try and go find it. And sometimes your cleaners are an extra pair of hands. So this is Steve back in the day with a shop vac, because we're getting the oil out of the ion pump when the service person was in to fix that. But wait, there's more. We're politicians, we're consultants, we're accountants, we're marketers, we're educators, we're mediators, we're facilitators, we're counselors. And thank you for laughing. So part of the reason why I structured my presentation this way was to say that a lot of the times you might not have like the biggest, the newest technology, but you have to actually work with the strengths that you do have and recognize the amount of work that you're putting in, even if other people are not realizing that's all the things that you do. So some of the other things that we do as a group that has a geologist and an engineer, chemistry, physics person, and a biologist. So we do cross-facility collaboration. So Douglas, he has received samples from the US, Iran, Egypt, Africa, South America, China, Ireland, Turkey, South Korea, and Argentina, because we collaborate with the other facilities at the University of New Brunswick. The geology polishing lab makes the thin sections. Douglas does the probe work and the analysis. The Mantis lab does the laser ablation, which is another level of analysis. So now you got a all in one place, one-stop shop. So lesson, be a one-stop shop, be one of the best, and also make sure everyone knows not just your field. So one of the things that I've done has been educating faculty members who kind of look at Douglas, because he's a geologist in the biology building. How is he advocating for the biologist? What is he doing there? He's always in this room. And I would be like, wait, no, you have no idea what he's doing. It's a lot, because of all the external clients that he brings in. Helps to keep the facility going. So I've had to correct that a couple of times. Other facility collaborations, so even within a facility, so these are coralline algae. They like to grow on stuff, and some of the stuff they grow on is rocks. And when you're trying to look at them, if you chip away at them, they kind of flake off and they're hard to look at. So our engineer was like, well, we tried using liquid nitrogen, it wasn't working so well. Why don't we just crush them in a vise? So we took these samples, we put them in little baggies, we crushed them with the vise, and that actually made these fracture points so that we could look at it in the SEM. And then when I was looking at these individual pieces, like, well, the rocks are actually very different from each other. So all of these 5 samples here are supposed to be different species. And it looks like they've picked different rocks. Well, maybe that's actually a part of the contribution to the difference in speciation. Well, good thing we have a geologist that we can be like, isn't this interesting? Let's do some analysis. And then the last example I gave was that even within your own discipline, taking your expertise and applying it to different areas. So a lot of scientists, they will only do experiments with the existing paradigm of these are the techniques that I'll use in my field, and this is how I do it, and away I go. So a lot of times, though, you can actually use techniques across model organisms, or even across different disciplines, like microbiology, immunology, that are actually different, to come up with new outcomes. So these are different sponge species. So we have 1, 2, 3, 4, 5, 6, 7. 7 on the left. The sponges like to grow on coral, and so that makes it very difficult for them to section. But if you use decal, which is used to decalcify bone, you can actually remove the coral, and now you can actually section them quite beautifully. So I bring this up as an example also for another reason, because this on the left was from 2014. And one of the things that people also don't realize is how much time and resources we save researchers. This paper originally came back with reviewer comments asking for SEM of all of these different species and these sponge cell aggregations, and when they come back together. And so with doing some histology, we can actually show, hey, that's a lot of additional work you're asking. And they're like, yeah, we didn't realize that was a lot of additional work. No, never mind, you don't need to do that SEM anymore. So that's how we got past that situation. For myself, it's important to recognize your own strengths. Sometimes I feel a little bit out of place with all of the specialization, but there is value in being a generalist because you can answer different questions and navigate people into appropriate directions for their research. Be of service to everyone because there's strength in numbers, so it's not about creating relationships within your facilities and your users, but also with other facilities too, and other staff members, because oftentimes they're your biggest advocates. And then communicate, because a lot of faculty members actually don't really know that we do all these things. So unless we have that conversation with them, they assume otherwise. So this was me back in 2014, training students. This was the lady with the blonde hair is Arlene Oatway, so she was my mentor, and the previous person of the microscopy facility that I used to work in, that I owe a lot to. And then I share this only just to remind people that, you know, we do a lot of science, but also we're connecting with people too. So I thought it was really funny. This was the retirement invitation for Gary, who started all of this, and you'll notice that you're supposed to bring his favorite beer. No nasty cards, but they will be tolerated. And then, so I just wanted to extend a thank you to my colleagues. Really, again, it's not really about me, it's about them. It's about the facility because they are awesome to work with and they make the facility what it is. Thank you to all the people at the aquatics facility at the university for their support in me as a professional. Thank you to Bina for the opportunity to do the core facility training course. So if you get a chance, I highly recommend that course. It was awesome. I learned a lot. The Canadian Federation of University Women for supporting my education. CNSP for these opportunities and all the advocacy. I think it's tremendously needed and is very appreciated. And also to Microscopical Society of Canada for supporting microscopy in Canada. Also, by the way, we have a meeting that's going to come up, so if you'd like to attend and see us, that'd be great. And in the end, all the machines go to the same place. So Any questions?
Are there any quick questions? Yep, we have one. Go ahead.
Fact that nasty. The fact that you are basically teaching a lesson to CFI, that we should not strive to get the latest equipment so that it can become, after a couple of years, a $10 million paperweight, but rather that equipment can be used forever, well, until it reaches that stage, of course. But so thank you very much. I hope that CFI can hear you.
Any final questions or comments? Okay, so thank you. And before we end the session, we have, I think, one tech bite. No? Okay.
So we don't have a tech bite right now, so we're good. And thank you to all the speakers today and the moderators and everybody for staying on time, for your great engaging questions. And we have our dinner tonight at Beatrice Restaurant, which is about a 25-minute walk from here. It's about a 15-minute walk from the Delta Hotel. So if you just go down University to Sherbrooke and turn right and just keep going, you'll find it. But there's a map in the program as well with the address there too. Tomorrow morning between 8 and 9, there's going to be light refreshments. So no bacon and eggs and toast, but there'll probably be coffee and juice and a few snacks. So if you don't have time to grab breakfast, there should be something here. And we'll start again tomorrow morning at 9 o'clock. So thank you.